C. Okay, nice. So on the C drive, IMBRC. All right. Just give me a minute to set the scene. Welcome everyone to the first session of the IMBRC Symposium 2026, diving into marine mines. I just want to present the jury very quickly to you. We have Florence Méjanet from Sorbonne Université, Nicolas Jegri from UBO, Belen, I already forgot your last name, Gonzalez, from here, the Marine Station in Plenzia, Idea Campora from ATU Galway, and I'll be chairing this session, and Olivier Gauthier from UBO. Just a reminder to all of you that are presenting in this session, you have 15 minutes to present your work, and then we'll have a 15-minute discussion. When you come to the 12-minute mark, I will slightly wave to try to see that. And if I get up, is that you need to finish.
All right. And it's okay for all of you if I put a little timer next to me?
Absolutely. If you need that to place yourself, please, by all means, do it. So, Simon will be opening this session for us. So, whenever you're ready.
Yes, this is the pointer. I'm going to check if it works. It's maybe too fancy for me. I will maybe use my phone.
Whenever you're ready, you can do it.
All right. I'll start. Good morning, everyone. I'm Simon Peters. Dear colleagues, friends, and jury, I'm super happy that I can present my thesis here for you. I'm honored to be the first one. My thesis is entitled A Flow-Through Mesocosm Study, Biochemical and Physiological Responses of a Three-Level Marine Food Web on Conventional Polyethylene, Compostable Plastics, and Tire Granules. Okay, let's start. As everybody knows, marine plastic pollution is one of the major problems of our time, very visible here. I'm going to demonstrate it with some numbers. Approximately about 400 million tons of plastics are produced globally every year. But to actually understand and highlight the importance of this study, I'm going to show you some other numbers. 4.8 to 12.7 million plastics are estimated to end up in the ocean. Once they enter the ocean, they just don't disappear, of course. They have very slow degradation rates. This is because of limited UV light and also lower temperatures in the ocean. This is particularly evident in the deep sea, where we have super low temperature and almost no UV light. So degradation rates are almost negligible. Finally, there is not a real solution directly inside because only 10% of the plastics are getting recycled up to this day. But now, one of the emerging solutions are biodegradable plastics. Biodegradable plastics, there are some certificates that are built on industrial settings for degradation, but they don't really represent the marine environment. Another major problem of our oceans are tire granules. They end up in the ocean from synthetic sports fields next to the ocean, or maybe protection sources at harbors. There's many ways that tire granules end up in the ocean. So yeah, this thesis was eventually motivated by different research gaps. Firstly, biodegradable plastics, they are marketed as sustainable, but still ecotoxicological data is inconsistent. Earlier, some studies have already shown, like Beiras et al. And Coade et al., that the toxicity rates might be higher than just normal conventional plastics. Secondly, there's a big research gap in the multitrophic approach in ecotoxicology. Almost no study uses a multitrophic approach. Why are they so important? Because hydrophobic chemicals, they can get transferred to different levels, so they're very interesting to study. Finally, Marthasteras glacialis has been earlier proposed as bioindicator around the coast of Galicia, but the biochemical biomarker data still remains absent. Let's dig into it. Okay, so how did we approach these research gaps? We started a flow-through mesocosm experiment with 12 tanks in total. In this mesocosm, we introduced a marine community of 3 different species. Firstly, with a primary producer, Zostera marina piscibras, and with a filter feeder, Mytilus caloprovincialis, the mussel, and finally a predator, Marthasteras glacialis. We used 4 treatments of 300 grams each in a 300-liter flow-through seawater. The treatments we used was tire granules, conventional polyethylene, biodegradable plastics, and finally, of course, a control. Okay, so here you can see the setup at the mesocosm facility of UVivo at the SIMAT. And to characterize all of our materials, we used Fourier transform infrared spectroscopy. This we use to identify the polymer out of which our material consisted. Then we did gas chromatography, mass spectrometry, to identify the additives that were present inside the materials. Now, for our community, Zostera marina, and here you can see how Zostera marina was mounted inside our aquarius in this little grid. We used photosynthetic efficiency as an endpoint. Here you can see the dark adaptation time of the Zostera marina leaves prior to the measurement. And we also used shoot length. Then, Mittelus provincialis, our filter feeder, the mussel. And here you can see a specimen of Mittelus edulis, which is actually anatomically equivalent. We used the gills, you can see them here with number 1, to look for enzymatic activity of the biomarkers acetylcholinesterase and glutathione S-transferase. Then, on top of it, we used the digestive gland, which is right underneath this muscle, for the enzymatic activity of glutathione reductase. You also have a picture of the gills after dissection. We also measured some physiological indices, like the condition index. Now, Martasterias glacialis, our predator, the starfish. This thesis has been quite novel because we didn't find any literature that proved some biomarkers like glutathione S-transferase and glutathione reductase in asteroids before. How did we approach this? We used the pyloric second, which is these yellow structures. Here you can see them after dissection. And there we looked for the enzymatic activity of these two biomarkers. Of course, here we also measured some physiological indices like the gonadosomatic index, and we also monitored the feeding rate. Now we got a whole lot of data, so how did we approach it statistically? We used linear mixed effect models, and they were particularly relevant because they could account for the repeated measurements of the starfish that we did, and also for the tank variability. Then our models were selected with likelihood ratio test and a Akaike identification criteria. Finally, we did some post-hoc comparison to compare in between the different treatments and time points, and we constructed some multivariate biomarker responses to the partial RDA and Peperamanova. So, now everybody is finally familiar with the full experimental setup. I'm going to introduce you to my hypothesis. Hypothesis number 1. We hypothesized that enzymatic biomarkers would be a more sensitive indicator of ecotoxicological stress than the physiological endpoints. Our second hypothesis, we expect that the biodegradable plastics and the tire granules will actually induce a biochemical stress response in the whole marine community. Thirdly, we hypothesized that there will be an enhanced biomarker response in Martasterias glacialis comparing to our lower trophic levels. And then finally, a higher chemical hazard, more chemicals, will also reflect in a stronger and a more consistent biochemical response. Okay, let's go over to these results. As I told you, we did some gas chromatography, mass spectrometry, to identify the additives in our materials. We actually found no real hazardous materials in the conventional polyethylene, and we calculated a chemical hazard index of 0. Now, when we go over to our biodegradable plastics, we calculated an intermediate chemical hazard index, with these hazard components as the most prominent ones. Now finally, with our tire granules had definitely the highest chemical hazard index, and the most prominent hazard compounds were like polyaromatic hydrocarbons and also a very emerging one, 6-PPD quinone. Okay, how did these materials affect the full marine community? That's what we're looking at right here. You can see Zostera marina, and you can see on the y-axis the photosynthetic efficiency measured by the pump fluorimeter over a period of time. This figure shows quite some noise because we got all the measurements of the different shoots, but we could actually not really see a treatment effect overall in the photosynthetic efficiency. The same goes for the shoot length. What we did see was very weird that we saw like a lower photosynthetic efficiency here and here. Luckily, we had some monitors of irradiance in our aquarium, so we mapped the irradiance on the background. When we can see increasing irradiance, we could actually see a decrease in photosynthetic efficiency, like here and here. After this would be recovered after some time, and this would be because of the change in pigments. That is what we hypothesized. So overall, we didn't really see an effect of our treatment in the photosynthetic efficiency of the seagrass. It was mainly due to other abiotic factors. Let's go over to the filter feeder, the mussel. Here on this figure, you can see on the y-axis the enzymatic activity of the biomarker glutathione S-transferase, and here are different time points. The different treatments are explained by the different colors. What we could find was in intermediate day, we actually saw an inhibition of the GST activity in the treatment with the biodegradable plastics. This contradicts our hypothesis where we were thinking of an induction. Eventually, we saw a recovery of all the treatments by day 5, and they all had very similar values. This recovery is probably due to the end of the leaching of the plastics. When we looked at our other biomarkers, with the diuretic and acetylcholinesterase, we didn't see any treatment effect, neither did we in the physiological indices. Okay, let's go over to the predator, the asteroid or starfish. Now, this is the same figure with the glutathione S-transferase activity, and we saw some very similar results. We saw again that the treatment with the biodegradable plastic was significantly reduced at intermediate sampling day. On the contrary, what we also saw was a significant induction of the tire granule treatment. And overall, at the end, the treatments again recovered through their basic levels. What is important to mention is that wet body weight was a very important predictor of the GST activity for our starfish. Now, similarly as in mussels, again in glutathione reductase, we didn't see any treatment effects, neither did we in the physiological indices. Now, for the last part of my thesis, I wanted to test whether these contaminants then actually propagate throughout the food web. And what was my approach? So, what we did was take the GST activity and we standardized them within the species. And then we compared them again with linear mixed effect models. What you could see for the biodegradable plastics, we could see an overall inhibition of the GST activity. And the effect was very similar in the two treatments. On the contrary, in our tire granules, we see a different response. In the mussels, we saw very small or almost no effect, comparing them to our starfish, where we saw an induction of the GST activity in the starfish. This is very interesting because it could be due to the different pathways that the starfish were actually exposed to the treatment. They were, like the mussels, exposed to the seawater in the aquaria, but they also were continuously feeding on the mussels, which were previously exposed to the treatment. We know that mussels actually accumulate chemicals, hydrophobic chemicals, like 6-PPD, and polyaromatic hydrocarbons in their tissue. So this was an interesting finding. We hypothesized that this could be due to trophic transfer. Now, what are the key takeaways for this presentation then? We can confirm our first hypothesis that GST that the biomarkers stay early endpoints for ecotoxicological stress. GST detected the first treatments and the physiological indices didn't show anything. They remain very important early warning indicators. Then, secondly, biodegradable plastics, we cannot consider them automatically safe. So, the biodegradable plastics, we could see that they produce the most consistent biochemical response, although they showed a lower chemical hazard index than our tire granules. So, we could actually reject the hypothesis that the chemical hazard index was very closely bound to the biochemical response. So, chemical hazard index alone is an insufficient predictor of ecological risk. Finally, trophic transfer really matters. This is one of the key points of my thesis that I want to give away. The TG response was exclusively seen in our predator, and this was, as I said before, consistent with the dietary transfer hypothesis of the hydrophobic compounds. If we use single species studies, we could have missed these results. Now, finally, we found some very promising results for the biomarker analysis in Martasialis glacialis, which showed similar to the mussels. Martasialis glacialis are asteroids. These are the first enzymatic biomarker protocols that are written for Martasialis glacialis, and in asteroids, the first time that glutathione reductase and glutathione S-transferase has been used. This supports its emerging role as a bioindicator. So, of course, I'm going to be honest, this study also had these limitations. The exploratory nature of the study made us use quite a high amount of plastics. On top of that, the tire granules were in mesh bags, and this limited their UV exposure a little bit. Oops. We didn't quantify the chemicals in our leachates, neither did we in the tissue. But, of course, these limitations are not limitations because they're just opportunities towards the future. Now we can start to quantify the chemicals that are in our leachates and also in our tissues, in the mussel and the starfish. we can start with some longer exposure experiments so we can maybe monitor what happens with these physiological indices. And finally, we can, of course, use some more environmentally realistic contaminant mixtures, especially with inhibition of that we saw in the bioavailable treatment, and some environmentally realistic concentrations. Finally, it would be very interesting to really construct a full battery for the bioindicator Martasterias glacialis. As I can show you here on the map of Europe, we can see the omnipresence of Martasterias glacialis around Europe. It's almost every coastline, and it's very easy to find. So it really supports its emerging role as a bioindicator. I would really like to acknowledge my lab and everybody who was there. It was an amazing experience. You've seen the huge mesocosm things that I used. It was super cool to work with them. So I would like to thank my supervisor, Dr. Ricardo Beiras, Dr. Leticia Vidal for the biomarker methods that she helped me with, and the diver, Roberto Gomez Alvarez, because it was not easy to sample that high amount of starfish. Thank you very much. I'm very happy to take any questions.
Thank you, Siemens. Very good use of the time. Thank you for this dynamic presentation to start off with today. I suggest, jury members, there's 4 of you. We start with maybe 3 minutes each, if you're willing. I don't know if you, and then we see what time we have left. Who's willing to start? You want to go ahead, Rose?
Oh yeah, why not?
All right.
So first I congratulated and I thank you also. I join in with the review. So I would like to ask you a question about your chemical index.
Yeah.
So you did the GCMS analysis yourself?
Yes.
Yes. And what was the target compounds? Because you gave us a little bit of results.
Yeah. This was actually done by the chemical team in Uvigo. So I know we sent our materials and they just checked in a list of materials which additives that they use the library to detect the materials that they could find in our samples.
And which additive, for instance, would you expect to find in tires?
In tires, we were mainly thinking about like the polymeric hydrocarbons,
Yes.
which we found. We also found a lot of studies that were proving like the emerging toxicity of the 6-PPD that can be transformed into ozone to a very toxic chemical, ketone. So these were the main ones that I was concerned for, and we also find them in our analysis.
Phthalates were targeted by the analysis?
I think so. I think if I can go back to the slides. No, I didn't put them here. Okay.
Okay.
Nice.
Thank you. You have another
Oh,
one?
yeah.
So it's interesting because you are testing some plastics that are really different.
Yeah.
Polyethylene,
Yeah.
the biodegradable plastic are
Yeah.
different. And the key environmental problems that you are targeted are the tires.
Yeah.
And do you know what they're composed of in terms of kind of plastic? How this plastic compares to polyethylene?
Yeah, we also did, like, as I told you before, the Fourier transformation infrared, and there we detected no real main polymer, but the results I can find in my thesis, but there was like, I think, a match with 3 different polymers that had like a 60% accuracy rate, so I cannot answer that, sorry.
Okay.
We have time for a quick last one if you want, Laurence.
Okay. And then you are giving us some new direction for the research. And it's like, okay, I believe that there is a link through the leachates towards the organisms.
Yeah.
And you plan maybe in the future, or you give us the advice to look in the leachate.
Yeah.
How would you do that if you had to plan like for PhD or whatever?
Yeah. Actually, this is a little continuation that we did, and we just introduced our plastics again in aquaria, and we took water samples, and we're sending them to the same institute to analyze for the chemicals that are in the because as you saw, there's a higher chemical hazard index, and this was surprising that we then saw a more consistent biochemical response, and this is probably due to not all the chemicals being released in the leachate, or there's more hydrophobic compounds that are less easily
Yes,
in the
probably
leachates.
yes.
Yeah. And they're mainly hydrophilic compounds, so yeah, they come anywhere very easily.
Alright. Who wants to go next? Kelly? Ellie? Alright.
Thanks for the presentation. I thought you made it really well understood. Excellent, well done. So the type of when we are looking at plastics in general, we are concerned about them being in the environment because of everything they cause and all of that. And with your research, what do you think you're getting? What are you adding to this?
I think it's just also an important view that everybody should consider, because you see the huge amount of plastics, and we're switching to biodegradable plastics, but it's just the same problem that we keep on reusing it back and throwing it away. So for me, it's very important to just use more durable things, and that we just don't use for single use. Because if we're just using this one and we're throwing it in the ocean and it's everywhere, and we're gonna switch to this one, it's not even convinced how quick they degrade, and maybe there is some toxicity that also goes along with it that we should definitely consider and not just throw out the window.
Okay. The thing I'm asking that for is because obviously everything they use were virgin plastics, so they don't really tend to contain all of these contaminants that are absorbed while they're there. So how do you think you could improve your experiment to take that kind of stuff into account? So plastics in the environment looks very different to what you have in the lab as virgin pellets. Does that make sense?
Yes, we could use indeed plastics that have previously been exposed, but now we started with fresh plastics. Indeed. If that was your question.
Could you talk me through the chemical hazard protocol? So you went through the FGIR and you were looking at the chemicals that weren't there, or the ones that were, so you could account for them when you're doing your depuration?
As I mentioned before, I was mainly in control of the biomarker methods and all of these methods, but this was mainly the chemical stuff that helped us with this analysis. I know some things about it. I looked it up, but I cannot go step
Can you
by
tell
step
me
on
what
that.
you know about it? So.
The Fourier transformation, what they did. We just looked for the main polymer with Fourier transformation, and just been it's a target analysis, and it's with the infrared light, how much light it absorbs.
But with the GCMS?
The GCMS,
Yeah.
yes.
No, but with the GCMS, so what was the actual protocol? So which chemicals should you not be worried about when you're doing your analysis?
Sorry, I'm not super clear on the question.
So you said you went through a chemical hazard protocol. So basically, you looked at all of the chemicals that were present in your virgin pellets.
Yeah, you mean these ones?
Yeah, you're looking.
Okay.
Yeah, so then you actually could know if they were there or not.
Yeah, so we got our list back from the chemical lab, and then we had our different compounds, and then we looked at the match and about the surface area that there was in our sample, and then we went to a library to look for the indices.
Okay. Is this a protocol that you followed?
This is a protocol.
Paper,
It's a paper from my supervisor, Ricardo Beiras, that calculates chemical hazard index. So it's a confirmed method that has been used before.
Thank you.
Alright. Another Nico?
Yeah. Thank you. I appreciated your presentation. Nice slides, quite clear. And I'm definitely not a chemist, so I won't annoy you with the chemistry part. But I have a question about the experimental design. How much were you involved into the choice of species and everything?
We were basically involved in everything. The diving sampling of the species was done by the diver. But we did the whole setup together with Melissa actually. We did the full setup of the cages, we introduced plastics, we built our experimental methods. That's of course together with our supervisor. I guess.
Okay. Because one of my questions is that one of the interesting novelty of the study is to have like this multi-trophic aspect. But about the choice of species, I was a bit. It was a bit weird to me that you choose, for instance, the seagrass that is not typically connected to the other one.
Yeah, I see.
And so, how do you think this impacts your result and your claim of having more multi-trophic?
Yeah. I thought that was also a very interesting point to look at if we could actually have a grazer of the actual primary producer. But we chose seagrass in the end because of its simplicity for sampling, and we also have it's also a very keystone species that has very important ecological implications. So that's why we chose seagrass and also mussels as a filter feeder. It was much harder to work with microalgae or things that could be transferred to the mussels because we had the open flow through. So like continuously putting in new microalgae would just flush them away. And if we would introduce new microalgae again, then they would not be exposed to the treatment from zero. So that's why we chose seagrass as a stationary.
Related to that, so you say that you see a trophic effect because you see different, you see more effect basically on the sea star than on the mussels. And I was wondering, how can you be sure that it's really a trophic effect, or just an effect of the different physiology of the?
That's also a very fun direction to go into, and that's why we really want to do the quantification of the tissues of our starfish and of the mussels after this project to give it some continuation, so that we could see an effective chemical transfer maybe in the different species.
Would it have been possible, I don't do much experiments anymore, but would it have been possible to include perhaps a control where you feed them non-exposed mussels, or something like that, so that you can see?
It would be possible, but I think they will always be a little bit hard. They will always be a bit exposed in this case because the treatments are just there in the aquarium, and they will always be a bit exposed. They can be less exposed if we introduce them fresh. Now every time I did some feeding, I put them first inside the cage for a few days so they could accumulate all the chemicals that are present in the water, and then I went over to the feeding. So maybe I could. That would be indeed an interesting thing to do, but we already have 12 aquaria, and 3 replicates is kind of limiting already. So yeah.
Right, thank you, Nico. Dylan, you have the.
Sorry, because I'm the last and I have been taking already all my questions. And I'm also working in analytical chemistry, so I may be dig a bit more on that. So one idea that Laurence treated a little bit is like, yeah, you had a target list probably with all your chemicals that you want to look if they are present in the plastics or not. Here you mentioned some that are present, so you positively, you or someone else positively detected, but did you have the chance to quantify that? So do you think that it's relevant how much of those is present or not?
Yeah, we had the surface area and percent of match of the chemicals.
But like the concentration, like is it the same if it's 1 nanogram per gram of
I
urine or
see what you mean.
100 nanograms?
Yeah, these were all chemicals that did have a significant presence in the samples.
Yeah, but it would have been nice, don't you think, to connect concentration with effects?
Yeah,
Because
yeah,
of course,
yeah.
normally ecotoxicology is is correlated with the presence of chemicals.
Yeah.
So that would be something to add or to think about. Also, I was you mentioned that with the mussels. Can you show the graph with the evolution? Yeah, in here. So you said that there was like an end of the leaching at day 5.
Yeah.
Do you really think that the plastics end leaching all their
No, they don't.
chemicals?
They don't end leaching, but there is some other papers made by our lab, and where we saw the most toxic effects of the leachate, especially of biodegradable plastics, in the first few days. They don't stop leaching, but the major important chemicals the first major effect was etched out.
Yeah, because for instance, I would expect, I'm not an expert in plastics, but I would expect like in 5 days, maybe the more hydrophilic compounds are released, but the hydrophobic will still remain longer. So then longer exposures are maybe more expected to release the more bioaccumulative chemicals and maybe the more toxic chemicals.
Yeah.
So it's a great idea that for the future you're doing longer exposures. I really support that. Also for the stars, you did say as well, if I didn't misunderstood, that body weight was really connected with the chemical index. How you?
No, with enzymatic.
Yeah, with
Yes.
sorry,
Yeah,
with the
yeah.
enzymatic. So can you please explain that better?
It just we measured body weight we constructed our model and we saw that the body weight was a positive predictor. So how much more body weight it had, how much more enzymatic activity we could see in the
So
starfish.
I would maybe assume that you kind of normalize your activity. So a bigger individual will have more, I don't know, more active metabolism or the opposite, I don't know.
Yeah.
But for me this relation, what does it add to the exposition and to the effects? I don't really get it.
It's just important to keep in mind when you do coming experiments that you monitor with body weight and you also include it in your modeling. You don't leave it out. That's just an important implication that I thought was very worthy.
Okay. And then maybe the last.
We have to
Okay, so.
Okay.
Thank you. Thanks a lot.
No worries, no worries.
Thank you to the DREAM members. Thank you very much, Siemens. Well done. So, congratulations. Next up is Elena. So Elena, you know the drill. I don't have to explain again. And whenever you are ready, you can take it up.
I will also put my name. Well, good morning, everyone, again. I am Elena, and I will be presenting to you my preliminary results of my master thesis work on the identification of potential links between ocean carbon sequestration intensity and microbial dynamics in marine snow, and more especially the role of diazotrophs in this process. During my thesis, I was the Institute of Research and Development in New Caledonia under the supervision of Sophie Bonnet within a project that's an European Research Council project, so within our vibes of IMDRC. In this project, we try to understand the carbon pumping by the ocean. So this is mainly done by the biological carbon pump, where CO2, atmospheric CO2 that is dissolved in the ocean is captured by the phytoplankton through photosynthesis and travels throughout the trophic web. When these organisms die, they will sink through the water column, carrying with them the carbon that was previously in the atmosphere. While sinking, some of this carbon will be remineralized, while another part will be going down deep in the water column, then being sequestered for the long term. If this mechanism wasn't working, atmospheric CO2 would be nowadays 50% higher. This biological carbon pumps works very well in nutrient-rich waters, but is not efficient at all in oligotrophic waters that are poor in nutrients, more specifically nitrogen. It turns out that in these oligotrophic waters, there's diazotrophs, an organism that is able to fix nitrogen from the atmosphere through a gene called EH. This nitrogen from the atmosphere is inexhaustible since it constitutes 80% of the atmosphere. And while capturing this nitrogen from the atmosphere, they become themselves an external source of nitrogen for the surface ecosystem. They are known to be the first source of external nitrogen in the oceans before the atmospheric input and river inflows. When becoming this external source, they also feed the biological carbon pump in these oligotrophic areas. This is known as the indirect export pathway. But recently, we've discovered that they also sink themselves, carrying the carbon that made them, being then a direct export pathway of the carbon. Both ways, indirect and indirect combined, represent from 50 to 80% of the carbon export in the Southern Pacific Ocean. So it's a very relevant process. In this map, I showed the chlorophyll contents, the chlorophyll concentrations at the surface of the ocean, and we can see in blue and in purple the areas of oligotrophic, of very poor in nutrients, so the oligotrophic areas that represent up to 60% of the surface of the oceans. And even some papers said that these areas are expanding up to 2% per year, showcasing how important it is to understand these ecosystems and the organisms that inhabit them. Mostly if they were actually very relevant, they made it very important to add into the ocean components of climate models for the future. In this thesis, we have approached this topic through through different gaps. So the first one is inspired from this time series in the marine station in Aloha in Hawaii, where they measured the carbon exported at 4,000 meter depth over 3 years, illustrating how dynamic this environment can be and how important these carbon exports can be at certain periods of time. But in this time series, they only had one time point every 15 days, missing out on the events on the short time period on high frequency on a daily basis. And also, they didn't know what was happening on the surface at the same time. So it is important, so we wonder if these high export events are actually constant or episodic in a high frequency timeline, and what was shaping the surface that could have driven this export. Secondly, the second gap is in the same paper, they quantified the diversity of microorganisms falling in the marine snow at that same depth. But again, they didn't know what was the community composition on surface that could have driven this diversity at depth. So we don't know if there are taxons in surface that are better exported than others, or if they undergo mineralization in a different way than others. So these two gaps have created the two axes of this thesis. First, we aim to understand the carbon export dynamics at high frequency, sampling simultaneously surface and depth. And then we want to link the microorganism communities from surface and depth to these peaks of export at high frequency. This project is called HOPE, and it located a high set of instruments along the Southern Pacific Ocean. For my thesis, I was using an intelligent buoy seated in the center of the instrument and a mooring line called export line. In this buoy, I was using a DNA-RNA autosampler that was measuring the surface water, as well as a CTD profiler of the euphotic layer. Combined in the same area, I was using data from the sediment traps located at two different depths, collecting the marine snow falling on the said area. Here are some images of the buoy, the DNA autosampler, the CTD cage, and sediment traps that I could manipulate during my thesis when I was on 2 missions aboard the RV Antia. Here is an example of 4-day sampling type of this project. In 4 days, we would obtain 1 sample per day on the surface. At 175 meters, we would get 1 sample every 2 days. And then at 500 meters, we would get 1 sample every 4 days. So in conclusion, on only 4 days, we would get 7 different samples, illustrating the genetics and the biochemical conditions of the whole water column at 3 different depths. So it's a very rich set of data. Myself, I was doing genetics in this sample. So I was taking the surface and depth samples and doing a qPCR analysis on the NIF-H gene, so characteristic of these diazotrophs. I wanted to understand the abundances of these organisms at different depths. And for the project, we are aiming for 3 different species. First one is a free-living that can be either filamentous or colonial diazotroph, Trichodesmium. And then other 2 that are using unicellular cyanobacterial nitrogen fixers that can be either free-living or symbiotic. These species are known to be either very abundant in oligotrophic areas or very important in nitrogen fixing. That's why we were aiming for them. Combined to these genetics, we will also get the carbon flux exported in the marine snow through collaborations with other labs. And finally, combined to the genetics and the carbon flux, we would also get the biogeochemical conditions in the surface through the profiling of the CTD cage that would give us the weather and the characteristics that were happening in the surface. For the second objective, we wanted to understand the community composition. So for that, we did some sequencing on the 18S and 16S gene on surface and sediment samples to actually understand the prokaryote and eukaryote communities at different depths of the water column. Here are some preliminary results of my work, because I will present it in August. We worked in a 2-month period time that was going from austral winter to austral summer, as seen in this temperature graph of the first 100 meters of the water column throughout the 2 months studied. At the same time period, we also observed the fluctuations of chlorophyll concentrations along the water column, and we can see that there is a high increase in chlorophyll almost reaching the surface around the 30th of November, showing a peak of chlorophyll at this time. Additionally, we also got the hydrographic context of the water column. In orange, we can see the wind speed that goes up to 25 knots in only 3 days' time and then persists for a long time. Combined to this, we can see in green the chlorophyll that we were talking about before, and we can see that it peaks only a few days after the start of the strong wind period. Additionally to this, we have in blue the mixed layer depth, so the mixing layer depth, that fluctuates a lot over this period, showing how dynamic an environment can be on a very short time period, only 2 months of oscillations from 10 to 60 meter depth. We can see that it reaches 60 meter depth around the same period of the chlorophyll peak and the strong wind export, maybe indicating that the wind made a mixing of the deep nutrients that fed the chlorophyll export at surface. Additionally, we also got the carbon export at depth. So here we have the particulate organic carbon exported throughout the 2 months at 175 meters, as well as the CN ratio, so the carbon and nitrogen ratios, at 175 and 500 meter depth. We can see that there is an increase in carbon export of over 5 times higher during the month of December. Additionally, we can see that there is a decrease between the CN ratio of 175 meters and 500 meters of almost 50%. It is important to notice that this increase in carbon corresponds to the same timeline only a few days after the peak in chlorophyll and the increase in wind on surface, as you can see here in red. Finally, we also got the qPCR analysis showing the relative abundances of the 3 diatom species at surface. We can see during this time period that there is a shift in communities where there is a predominance of leucine during winter season that shifts to Trichodesmium, which is a species typical from summer when entering the summer period at the end of the 2 months. A preliminary discussion of this first objective is that we can see that this sample period shows a very dynamic environment. Well, shifts happen in a matter of days, and then there's a cascade of events that we can see throughout the water column. A striking result is that the 50% decrease in the CN ratios at the 2 different depths. In literature, I've seen that remineralization tends to consume nitrogen first while the particle sinks, but we have observed the opposite. We have seen an enrichment of nitrogen while falling. So we wonder if there was a preferential consumption of carbon over nitrogen, or if there was maybe the high diazotrope concentration on surface that would have over-enriched these particles. So this is all hypothesis that we're working on right now. For the second objective, we wanted to understand the community composition at surface at depth. Here is the 18S sequencing, so the number of reads at surface and depth over the same period of time for each depth. At first glance, we can see that there is a strong decoupling between surface and depth communities. At surface, we can see that there's the typical surface communities that are photosynthetic or diatoms, for example, that actually disappear at depth, illustrating maybe a strong remineralization process that prevents these cells from reaching depth because they are remineralized. But not all follow this pattern, where Dinophyceae, for example, we find it at surface and also at depth. So there's different preferential remineralizations. On the prokaryote side, we can see also this strong decoupling, and we see something very interesting during the export time period that I was talking about previously. Here in red, we can see the time where there was a big export at depth, and we can see that there's a shift in communities of prokaryotes where there was a strong diversity that disappears, where some species become very abundant. So maybe showing us specialization about remineralizing new unexported marine snow that had just fallen throughout this peak of carbon export. Preliminary discussion of this second objective shows that we have seen weak variation of communities over time, but a strong variations over depth. And there's a strong decoupling between surface and depth in both prokaryotic and eukaryotic communities, showing how important it is to surface both surface and to sample both surface and depth, because it's impossible to understand what was happening in the depth by only sampling surface, as they are so decoupled. Preliminary conclusion of the first objective would be that we have observed a peak of carbon that could have been explained by two phenomena. The first one would be that we observe a strong wind that could have led to a mixing of the photic layer, bringing up nutrients that would have increased the chlorophyll at surface. And additionally, we also have a shifting in the season where there's heating waters that provoke a shift in diazotroph communities, leading to bigger cells that have more chlorophyll, like trichodesmium, that on the one hand, they have more chlorophyll, and on the other, they could have fed with nitrogen, also the carbon pump, so both leading to a peak of carbon export that we have observed. For the second objective, we conclude that this is the first time that we sample surface and depth simultaneously over a high frequency period. So we have observed that it's impossible to predict what's happening at depth only by sampling surface. So this is very interesting information. Both in eukaryote and prokaryote communities, we have seen a strong decoupling. In eukaryotes, we saw that depending on their the events that were happening to them, they can either be falling and we find them through the water column, or be remineralized or even advected by currents. For prokaryote, we saw that it was more dynamic and that there were shifts in communities also over time when a big export flux appears. These preliminary results have shed light upon the role of diazotrophs in this carbon pot, but there are still many research to be done, and there are still many questions, such as if there's predominant groups that are actually shaping the marine snow throughout these very strong carbon peaks, or if the direct export pathway contributes importantly annually, just not only 2 months, but wondering how does this how important it is throughout 2 years, or if it varies seasonally, or how much of this diazotrophic nitrogen comes from the direct pathway, or if vertical advections actually have an influence in this diazotroph-driven carbon export. And more on the technical part of this project, having these high-frequency datasets of several depths make us wonder if it's possible to have these datasets autonomously, so with this intelligent boy from the project, or if eDNA that is falling throughout with the marine snow, that is very broken and very fragile because it has fallen throughout many days until 500-meter depth, if it's possible to better extract it on the molecular analysis to get better signals of these genetics at depth. So these are all projects that are worked on in the group, and they will be discussed further on. So thank you very much for your time, and I'm open for questions.
Thank you, Elena. As you know, you were a tad over time, but that's not completely bad. We're going to start with the jury members. Belen, you want to go first this time?
Yeah, okay. Thank you. Well, it's congratulations. This is super nice work, super interesting, and also congratulations for being in such a place. I guess that it must be super cool. Well, first I was a bit missing a bit more. I know you have no time and you have preliminary data and you have a lot of data, super interesting to debate. But on the water columns conditions, so how deep is the area and how are the currents? And can you maybe give us some hints on that so we can understand better maybe how is really working the pump in there?
So you want the conditions of the
of the water column that you are evaluating, because I don't know how deep it is, if it's really in the middle of nowhere or if it's very close, because you showed a picture, so maybe it's not in the middle of nowhere. It's connected to some archipelago or
It is located outside, so there's New Caledonia, an island that has a coral reef barrier, and it's located outside of the coral reef barrier. So it's far enough so that we consider it open ocean, but it's not in the middle of nowhere, because we also need the instruments, they are put on, they're attached to, well, not attached, but they have a weight. And for example, the mooring line, 700-meter depth.
Okay.
So it is quite deep, but we have the instruments at specific depths.
Okay.
So yeah.
So it's 700 is the deepest, and you are measuring that your deeper area is 500.
Yes.
Okay. So how do you think that resuspension or sediment connection could influence the export?
Sediment resuspension, I think it is too far from the
Yeah.
sediment troughs to actually influence, because that means that there's a super big mixing of the
yeah.
layers. So I think it's everything that we collect on the sediment troughs, which are the instruments that are deeper down, I think it is influenced from the surface. So I think it's mostly currents that could influence.
Mhm.
So maybe the big bias would be understanding what's happening around that we're not measuring, that could put in information in our samples, but that came from somewhere else. I think that's the biggest influence, but sediment trough, I don't think.
Yeah,
But
and maybe
sediment-
just coming from the coast and know how close you were.
We were far away. We were outside of the coral reef barrier, so there was no influence from the coast.
Okay, nice. Regarding so in the traps you were only measuring carbon, but wouldn't it also be interesting to measure nitrogen?
Yes, definitely. Yes. That's what we have isotopic analysis. So yeah, that is part of the work to be done later. Well, we already sent the samples, but we're gonna get the results later on. But yeah, understanding where did that nitrogen come from, and understanding if it came from just a normal carbon pump or actually from diazotrope-driven nitrogen. That's something that we will do with isotopic analysis and in collaborations with other universities from Texas.
Yeah, it's topic. Yeah, it will give you a lot of information.
Yes.
One last one.
Okay, sorry. I could be talking about this.
No.
So the last one would be were you able to identify some deep chlorophyll maximum in the area, and maybe would that also be an interesting layer to sample or to figure out what's going on there?
Yes, definitely. It would be interesting. I haven't actually checked out that, but with the mixed layer depth, for nutrients, we were mostly interested about nutrients, and we have the profiling of the CTD. So that definitely tells us what's happening on the first 100 meters. But I believe since it's oligotrophic waters that sometimes it skips the 100 meters. So we don't know at which depth there's the chlorophyll maximum.
But also some blooms could be occurring at high depth, so you could be missing some interesting pumping at certain layers.
Yes, definitely. But that's one of the limits. But yeah, the blooms, we can definitely see them at surface. And we have seen that the water goes brown. So you can see blooming happening very often over there.
So nice, Elena. Thank you.
Thank you.
Thank you. Nicolas?
Thank you, Elena. I really like to talk as well. A lot of information. I had a question about so you basically see an event at the surface, and then you see this at least what you interpret as a reaction in the deeper community into prokaryotes.
Yes.
And I was wondering, do you have a delay between those two events, and how much is it?
Yes, we have a delay, definitely, because this is arriving around, the carbon exponent is arriving around December. And we have the peak of chlorophyll at surface end November. So there is a delay of several days. I haven't studied exactly how many days, but they're not synchronized in time, they're decoupled, which makes sense because we would expect marine snow to take some time to fall. And that's also something that is worked on the project, how fast can they sink?
Yeah.
And how much delay we would expect so that we're sure that we're coupling surface and depth.
And so that delay is compatible with what you know about the sinking speed,
Yes.
more or less.
Yes. It is not out of the normal.
Yeah. Okay. Because when you work at one station like that, the big issue as well always is that am I seeing like a time effect, or am I seeing like just like another water mass or something?
Yeah, that could happen, but we have ATCPs all around the study area.
Okay.
So measuring the currents to make sure that what we're interpreted as coupled surface and depth, it is actually true. So that's measuring current, and if it's under 12 centimeters per second, that means that's accepted by science as coupling surface and depth. And if it's over that, then we don't study it because it would mean that it comes from somewhere else.
Okay. I guess you have one more, a little like a simpler technical question. So you have like a buoy,
Yes.
and then there is a moving line where you have the sediment traps.
Aha.
Those are two different places, or are they far apart? Or?
No. they're very close by. We can do everything by boat in like 30 minutes.
Okay. Like
It's-
1 kilometer by.
Yeah, the whole area is isolated and there's all the instruments around, and they're very close by. They're supposed to represent the same area, so
Yeah.
they're as close as possible so that they don't touch each other and get entangled because there's currents that could.
And then, I guess a last more general question. So you said that there is some decoupling between what's happening in the surface and what happened at depth, and of course this is like in your communities, this is the main structuring factor. But still, you see there is a strong connection between what happened at the surface and what happened at depth. And so I was a bit surprised by your claim towards the end that you cannot understand what's happened at depth from looking at the surface, because there is still a connection, even if things are different. So I don't know if you can elaborate a little bit on that.
Yes, maybe I explained myself wrong. I was saying that if you want to understand depth, you need to also sample a surface at the same time.
Yeah.
And by only sampling surface, you cannot understand what happened underneath, because there's so many processes along the way that even if you had some communities at surface, what you will find at depth could be very different. So it is important to couple both, and just having one or the other could not explain the other one.
We're gonna need to move to the other jury members, so Heidi or Laurence.
Yeah. So, congratulations. It's very exciting. I have like thousands of questions, so I will control
Please,
myself.
2 minutes.
Okay. So about this precisely the coupling between on the different communities. When you look at this slide and the next one,
Yes.
you see, for instance, in this one, the shift in community during the peak of
Mm-hmm.
during the peak of the high flux events, let's say.
Mm-hmm.
How could you explain that, thinking or try to explain, thinking that if you are more organic carbon, maybe you favor more suboxys niche inside the pellets? Do you think that, for instance, this can have an impact on the carbon over nitrogen?
Yes, I think yes. I observed that these communities that were very abundant were mostly anoxic. They worked in anoxic environment and they were sulfur reactors.
Yes.
So that exactly. So that shows that this marine snow was very old and that had anoxic environment for these bacteria to be there. And this changes when there's new carbon arriving that hasn't had the time to become anoxic yet. So that definitely changes how the particle is remineralized, because they have different strategies for eating up the nutrients inside the particle. So that could definitely affect the CN ratio. But it's very strange that there's more nitrogen still, because the nitrogen is the main element for being able to while remineralizing. So it is a limiting element. So
Precisely, if you have bigger export, like maybe you have bigger particle, maybe the falling rate increase, and then do you see a connection, like maybe those anaerobic niche don't have the time to develop so much. You don't go towards the point where you have sulfate reducer.
Yeah.
Maybe you are still in, I don't know, nitrate oxidation, anamorphs, etc. So maybe that's something that you can make hypothesis with that.
yes, definitely, I agree.
So yeah, I think it's a real change. And also with if you look at the for the eukaryote, it's very nice to see the difference between 4 and depth, let's say.
Surface and
Yeah. Do you have an hypothesis on why there are some algae that are selected by the export? What is the mechanisms for that?
You mean that the photosynthetic one that would be found also at Mesopelagic?
Yes.
That they're not remineralized as the first. They're not the first option to be remineralized when being in the marine snow. I think that would be my first hypothesis.
And under which form they are on your marine snow? Did you have a look under the microscope on what was there?
You can see just agglomerations of white matter. So if you look at the microscope, you can also see what the marine snow is made of. So
Yes.
sometimes it's copepod carcase, or sometimes phytoplankton agglomerations like this, or pellets
Yes.
from zooplankton. So yeah, that's another branch of the project, understanding what the marine snow was shaped with. So that could definitely, if they are found in deeper and down, that could be
But for instance, here, can you make an hypothesis? Like, I would dare give you one. on the composition of the marine snow when you look at the specific export of those dinoflagellates, for instance.
Yes, I think it's definitely zooplankton material, because seeing that there was a lot of phytoplankton developed at surface, and there are some species that are zooplankton related, I think it's definitely a product from the grazing that will happen on surface, and then the carbon was exported through the grazing, so the fecal pellets and the carcasses from zooplankton. So yeah, I think that's more like zooplankton results, more than the phytoplankton exported itself.
It's like, let's say, you can make the hypothesis, you would make the hypothesis that specific grazing on those dinoflagellates would create fast sinking pellets that you will collect.
Yes, definitely.
Right. Thank you, Laurence. I'm sorry I need to cut you off. I know we could go on forever.
Yeah.
You can do that at the break. Heidi, a very short question or comment, please, because I was bad at policing the others.
Okay, sorry. Thanks very much, Elena. That was very well done. I just kind of wanted to be a bit more general. So, how does your research actually contribute to like climate change research? Since obviously this is a very important topic.
I think what we're aiming is to understand if actually these diazotrophs help exporting carbon much more than what we know nowadays. And so if they're important, if they're that important, that would be added up. So their role, their export, their influence in this carbon pump would be added up in the models for predicting the climate, like the evolution of the ocean. So the climate models, for the future. So I think it is, as we're adding up other organisms that influence, we are always feeding these models to be as realistic as possible. So the goal is to actually understand if these ones deserve to be added up because they have global importance. So yeah, I think if they are important, that could shift a lot our understanding of the carbon pump in the ocean.
All right, thank you very much.
Thank you.
Giada will be closing this session.
This
When-
is the clicker.
This is the clicker. You can get used to it if you want. And there's supposed to be the pointer with it. You didn't. Okay. So whenever you're ready, the floor is yours.
Good morning, everyone. My name is Gianna Payton. Thank you so much for being here. And I'm going to be presenting on the spatial and temporal variability of Honduran benthic communities and reef functionality throughout the 4th global coral bleaching event. So starting with a brief introduction, what exactly is coral bleaching and how does it affect reefs? So coral bleaching are symptoms of stress-induced climate change, and it's typically caused by prolonged high temperatures, which result in heat waves. So if this occurs across 3 tropical ocean basins over a common period, this is known as a global coral bleaching event. And the most recent event occurred in 2023 to 2025, and it's known as the 4th global bleaching event. So bleaching typically causes the loss of the symbiotic algae within corals, which leads to declines in their calcification and energy levels. And this results in phase shifts. So with our bleaching, we lead to declines in coral, which you can see here, with that 3D structural complexity, and also increases in fleshy macroalgae. So the decline of our structural complexity means there's going to be less habitat for fish and other species, so we're going to have reduced biodiversity and ecosystem services. So why are we focusing on Honduras? So Honduras is part of the Mesoamerican Reef, which is the largest reef barrier system in the Atlantic Ocean, and it supports high coral cover and ecological functioning. So the two locations we're focusing on have really high fish species, over 500 species, and 73 species of corals. So not only do they have really high biodiversity, but they're also really important for reproductive locations, for fish spawning, such as groupers, but also corals as well. They're also really important for coral donor sites for local restoration activities. So not only are they ecologically important, but they also provide a lot of economic benefits for Honduras. So fisheries support around $99 million annually, and tourism around $163 million. Unfortunately, these reefs are faced with a lot of increasing threats, so globally through these marine heat waves, but also locally through diseases, declines in water quality, and human impacts. So the research questions we're going to focus on is how exactly these benthic and coral community changes are occurring over time. And then we also wanted to compare the two locations and seeing if there's any variation in their response. And then finally, we wanted to look at coral communities, so seeing how the diversity and functionality is changing. So moving into our methodology and study areas. So like I mentioned, Honduras is part of the Mesoamerican Reef, which you can see on the left-hand side. And our first site is Tala Bay, in our top middle here, which is located on the mainland northern coast of Honduras. And we had a total of 8 sites here. And then on our right top-hand corner, we can see our second location, which is Roatan Island, and it's located about 50 kilometers north of the Honduran coast. And we had 3 sites here located on the southern point of the island. This is known as Cordelia Banks. So we had a total of 11 sites. So moving into our data collection, like I mentioned, this event occurred from 2023 to 2025. And it was sampled 3 different times throughout this event. So early stress period, which we'll see in the blue, mid-stress we'll see in the purple, and green we'll see is prolonged stress. So the protocol was done by Atlantic Gulf Rapid Reef Assessment, so it's a very standardized methodology, and it was collected with the Coral Reef Alliance and Healthy Reefs for Healthy People, as long as like local co-managers. So divers went into the water and placed 10-meter long transects, and they had a total of 100 points along each transect, which were spaced 10 centimeters apart. So each diver would identify what was under the point as the benthic substratum. So again, this resulted in 100 points per transect, which you can easily make a percentage from, 6 to 8 transects per site, and a total of 205 were analyzed. So our data collection is focusing on the live coral cover, which was identified to either the species or genus, Christos coralline algae, turf algae, and fleshy macroalgae. So again, these are the points each diver was identifying, and these could be made into percent coverages. So we focused on these 4 because they typically dominate the benthic categories, but also they're really good indicators of disturbance. So we also wanted to go one step further, so looking not just at coral cover, but also how that coral is contributing to the surrounding reef. So we did this by calculating a reef functional index. So like I mentioned, this is coming from our in situ live coral cover data, and then also looking at functional coefficients created by calcification rate, rugosity index, and mean coral height. And those 3 are coming from previous literature in similar regions. So moving into the statistical analysis, we started with a univariate analysis. So this is basically looking at how these individual variables were changing. So initially we looked at how they related to each other, and then we calculated coral diversity indexes for each site, and then linear models were done to assess how each period and location were affecting each variable. And then finally we looked at estimated marginal means to have an understanding of how exactly each variable was changing. And then we moved into a multivariate analysis, so we used dissimilar matrix, non-metric multidimensional scaling plot, and Permanova to basically understand how each community was changing as a whole. And then finally, we conducted the SIMPER analysis to see which variables were driving these community changes. So moving into our results, we're first going to look at live coral cover on the bottom here. As we can see, the first period is indicated in blue, the second in purple, and the third in green. So we can see between Roatan and Tela Bay, there's a significant difference in the T1 time period, with Tela Bay being slightly higher. But over the bleaching event, both locations had a significant decline in coral cover. And then if we come to our top right at the fleshy macroalgae, we can see a significant increase in fleshy macroalgae throughout the bleaching event. Then looking at our turf algae, we can see it had an initial increase and then a decline following our third time period. And we could see between Roatan and Tela Bay, there was a significant difference in how turf algae responded in the third time period. Then if we come to the top left, we can see Crusoe's coralline algae, and here we're seeing a bit of variation between how Roatan and Tela Bay responded. So overall, we're seeing many of the drivers are being driven by temporal variation, but there's some slight temporal variation. So moving into our changes in overall community, first looking at this plot on the left-hand side, again, T1 is in blue, T2 is in pink, and T3 in green. So each point here is representative of a site and the overall benthic community at that site. So first looking at the T1, we could see all the sites are clustered closer together, which is essentially meaning that they're more similar. And as the bleaching event progresses, they become more spread out, meaning that they're becoming less similar. And then if we look at the second plot on the right-hand side, on our y-axis is showing our benthic variables. So you have coral, fleshy macroalgae, turf algae, and Crusoe's coralline algae. And on our x-axis, we're looking at basically how each variable is contributing to the dissimilarity within the sites. So from T1 to T2, this first panel, we can see that coral is our main contributor. And then from T2 to T3, our last panel, we can see that the algae, these two algae, are contributing to the dissimilarity. So then moving back into our coral community, again, this is a very similar plot, each site as represented by a point. And then each one of these codes represents a coral species. So again, we can see in the blue, the sites are clustered very closely together, meaning they have similar coral species compositions. But as the bleaching event progresses, they become more spread out and more dissimilar. And then again, on our right-hand side, we have our y-axis of our coral species. And then along the x-axis is showing which one is contributing to the dissimilarity. So we could see across all three of our time periods, Agricia tenaphalaria and Agricia agricides were main contributors, as well as Porites asteroides in this last period. So then moving into refunctionality, again, this is just basically assessing how well the corals are contributing to the surrounding ecosystem. So again, we can see here there's a significant decline in refunctionality at both locations throughout the bleaching event. And then more importantly, we wanted to go one step further and look at how species richness and refunctionality interacted with one another. So we actually found a significant interaction between species richness and refunctionality index. And as we could see, there's a positive relationship between the two. So as species richness declined, so did our refunctionality index. So moving into our discussion, basically we found some indications of a phase shift since we have increases in fleshy macroalgae and a short-term increase in our turf algae. And we also had significant declines in our live coral cover, but this is also indicating that since we had declines in diversity and refunctionality, that it's maybe an overall effect on the ecosystem and not just short-term impacts. So again, since this was measured over 3 different time periods of a long bleaching event, these are long-term results and not just short-term impacts. And also these patterns are consistent with previous studies in the Caribbean and Mesoamerican reef, specifically on this bleaching event. So moving into our spatial variability, so like I mentioned, many of the main drivers were temporal variation, but we also saw some spatial variability between our sites. And this is likely driven by local environmental conditions, which may be different at each location. This includes things such as wave exposure, nutrient availability, and turbidity, and possibly even oceanographic conditions. So these differences may be mitigating the bleaching impacts between these two locations. And then looking at corals specifically, we saw some variation between the coral species at each site. So this could be due to specific coral colony adaptations or behavioral strategies, which may have occurred from things they've experienced in the past. So this could happen from like previous reef conditions, previous bleaching events, but this is known as ecological memory, and it could possibly shape some site-specific resilience to these events. So then moving into our overall coral community changes. So again, dissimilarity was driven by Agricia species, varieties, and some Acropora. Agricia and varieties are both really common species within the Caribbean, and these two locations used to be characterized by really high percent coverage of these species. But unfortunately, this shows just how extensive this bleaching event was. That's something that was really prevalent is now not. And then moving into our reef functionality, which also takes into account our coral percent cover. This means that maybe some of the changes we're seeing in reef functionality index are actually due to declines in our coral cover and maybe not so much as structural complexity. And then also just a reminder, we saw that positive interaction between species richness and our reef functionality index. So this indicates that maybe sites that have higher species richness may actually be able to mitigate some bleaching resilience. And essentially this means that corals that have higher species richness may have different responses to the bleaching event. So essentially different coral species have different abilities to respond to these events. So moving into our conclusion, again, we saw indications of a phase shift, so declines in coral cover and shift towards algae dominance. But our impacts extended past just coral coverage, but also affected diversity and our reef functionality, and also possibly our 3D structural complexity. And then some spatial variability indicates that locations may have specific environmental conditions that are helping mitigate these responses. So it could be really interesting for future studies to look at maybe water quality or oceanographic conditions at these locations to see if that's influencing their variation. And in terms of local management, we think that we should focus more on sites that are more resilient to bleaching events and understanding exactly how they're resilient to these events and focusing on how that can be applied elsewhere. So I'd really like to thank my supervisor, Antonella Rivera, and these organizations, Atlantico for Rapid Reef Assessment, Coral Reef Alliance, Healthy Reefs for Healthy People. Again, they collected all of the data, and the study wouldn't have been possible without them. So, thank you very much for your attention.
Thank you, Gianna. Well done. A bit under 14 minutes, so you had some time left. So, who would like to go first? Well, you can go, of course.
Gianna, thanks very much for your presentation. Well done. Very calm and well explained. So I don't know if I missed that, but could you show me how your time periods were divided?
Yeah, it's a little bit tricky. So like I mentioned, this data was all collected in 23 to 2025. But as we can see here, this is where we use satellite data to basically help identify when these surveys were going to be conducted. And obviously you can see here, this is the first one for Tela and the first one for Roatan. So they're not exactly occurring in the same time period. But they're using satellite data to basically understand when the thermal stress is going to be highest and when they think bleaching is going to start happening at that location. So it's not occurring exactly at the same time, even though the sites are quite similar.
Do you think that could be biasing your?
There's a lot of work into basically understanding how these events are hitting each location. So the sampling is very heavily reliant on this. So people are doing a lot of work to basically make sure that that's not a factor.
Then you were mentioning the ecological memory in terms of resilience. From the previous bleaching events that happened, do you have information on how these same sites had reacted to them?
Not specifically, or are you asking if they've experienced ecological memory thing? Yeah, there is some theorizing around that. Both of these sites have actually have higher coral percent coverage going into this bleaching event. So they think that maybe there's some sort of mitigation, whether that be local environmental conditions or ecological resilience and memory. So it's still kind of undetermined, especially since these bleaching events happen so close together. The last one ended in 2017 and this one began in 22. So the reefs don't even really have a lot of time to recover. So maybe it's ecological memory or maybe it's some other environmental factors.
Or was there any study done on the previous events or not?
Not specifically for this location.
Not for these locations, so yours is the first one. Okay. And then lastly, what will be the consequences, and that's what's basically happening to most of the coral reefs of like the extinction of flashy macroalgae taking over?
Yeah, essentially it's going to alter like your community structure as well as your fish. That's something we thought about maybe looking at in the study, but ran out of time. But initially there's a spike in herbivores because there's more algae. But then obviously the whole trophic cascade of the fish are thrown off as well. So then there's not enough herbivores and other invertebrate species to keep that algae in check. And then also the algae is going to be limiting coral recruitment. So the corals don't have substrate available to settle on. So there's a lot of spatial competition between the algae and the corals. So ultimately the algae will prevail if there's no other way to overcome their prevalence.
All right. Perfect. You managed to do 3 minutes perfectly just on your own. Who wants to go next?
I can go.
All right. Thank you, Madam.
Okay, so yes, connecting to, well, congratulations. I agree with your very nice pace and very good explanation. And connecting maybe with some of her questions, I was wondering on if you have considered, or anyone has considered about the recovery periods, because I think that could be also playing a role, because it's not only the memory in terms of which species remain, but until when and how, like the temporary, like the time they get to recover. Because you have been mentioned that macroalgae is maybe occupying some space because probably coral is growing slower or settling times is lower. So how can you maybe talk a bit about recovery and recovery times and how would that be evaluated if it hasn't been done before?
Yeah, it's been previously evaluated in some other different global locations, but there's a lot of obvious variability in those like results. But they think that it would take around 10 to 20 years for a reef to even recover from a bleaching event. And again, kind of speaking on the coral to fleshy macroalgae balance, like some corals obviously are slower growing or more susceptible to bleaching, so that's obviously going to be altering the coral community structure when these periods are so small.
Mm-hmm. Because for instance, in the graph you were showing with the T1, T2, T3, if you can go just fastly, which is the time variation between them. So for instance, in the one with the black, the next one, that one. So which is the difference between T1, T2, and T3 in time?
So again, that's kind of coming back to that figure that I was just explaining. Yeah. This one. So it's varying between locations, right? So our first from Tel Aviv, between the first and the second were only 3 months, but then Tel Aviv was sampled again here. So this was almost 2 years later.
2 years.
Mm-hmm. And then for Roatan, it had a bigger gap between the first and second. So this was occurring in June, and then again in December.
Okay, because looking at the comparisons between T1, T2, and T3, the main change is between T1 and T2, obviously, and then between T2 and T3, there's only slight changes for some species. So what I realized is that for Agaricia was the coral species. Is the fastest change, right? So yeah, between T1 and T2, the biggest is for Aten and Agar. Yes. And so do you think that is it really relevant to identify which are the most sensitive species and how could those impact the general ecosystem? Or because you mentioned Agaricia at the end that is one of the most relevant species. So if they are the most sensitive, as you can see here, is that going to be relevant for the ecosystem in general, right?
Yeah, I think this result with Agaricia is actually something kind of surprising. So previously, they've bleached really fast and really severely, but they prevail. They don't necessarily have mortality. But what we're seeing in this event is that they're actually having a higher mortality and they're bleaching. So they're not as resilient as they've experienced this multiple times. So it's kind of accumulation of these bleaching events. So I think it's still important to kind of consider all the corals and how they each contribute to the community.
Thank you.
Thank you.
Laurence or Nicolas?
Yes. Thanks a lot for the nice travel to these wonderful waters that you worked on. I wanted to ask you a few questions on the sampling. Was the sampling done always by the same diver?
No, there is diver variability, but this organization, Atlantic Rapid Reef Assessment, they have very highly trained divers who are trained on all the species. Like, it's a very intensive process.
And how do they check the quality of the data that the divers are collecting? How they
Yeah, I think
if they do like specific procedure?
it's mainly from the training. Like, most of these people are actually scientists as well who work with the data, so they're familiar with it. But yeah, it comes from like a big intensive training.
Could you think of some statistic on some mock dive, for instance, to check on the variability between divers?
Yeah, possibly a Permanova since it's multivariate. But the thing is, there's also community variation as well, so you wouldn't necessarily know if it's coming from like the diver's subjectivity or if the communities themselves are different.
In the literature, you have seen some results that are coming from this kind of observation and how they assess this issue, for instance?
Not necessarily in the literature, but ACRA has been around since the early 2000s, so they've had kind of a lot of time to progress and work on this methodology.
And I have similar questions on the RFI that you calculated also. Is it something that is like the functional coefficient? You get it from the literature. Do you think that this coefficient is stable along the bleaching, or what is the effect of bleaching on the coefficient?
I think the coefficient is taking into consideration calcification rate, rugosity, and mean coral height. So I think these are metrics that are not necessarily going to change in such a small time period. I think what's going to be changing more is the percent cover that's affecting the overall functional index.
Okay.
Thank you, Laurence. Nicolas?
Yes, thank you. I guess I had a question about the a bit more on the spatial side, because one of the big effects you see if you go back to your NMDS plots. Maybe not this one, but the coral one. Yes, on this one it's pretty clear. Like, and you mentioned that like one of the big effects is that you see an increase in your community variability when you get into the perturbation. And I was wondering if you thought about putting that in terms of like a beta diversity perspective, and what would it mean, and how would you interpret that?
I'm looking. Sorry.
Like, if you know there is like different component of community diversity, alpha diversity, beta diversity, and you mentioned species richness, for instance, which is alpha. This is more beta diversity, and it seems to increase with time. So, have you thought about that and what that means?
Yeah, we did also look at a few other diversity metrics. Here. So we have species richness, diversity, and Pallot's evenness. So we can see kind of that there's like an overall decline in all of them, but there was a little bit of site variability between these locations as well. So again, I think that's just coming back to something is happening within the sites that are kind of affecting.
Yeah, but that's all alpha. That's not beta. It's fine. It was a bit of a notebook anyway. I had another question. So you find this relationship between the functional index and richness, and I don't know anything basically about this functional index, and I was wondering, do you know if there is a way in which this relationship could be built in the way you compute it, or it's really independent and you can be confident of the relationship you found?
Yeah, we ran a linear model so to see that the species richness did have a significant impact. So.
Yeah, but like on the way you compute the functional index, maybe there is a way in which richness is somewhat represented in there. I'm not sure. I don't know it.
As far as I know from literature, it hasn't been found yet. This free functional index is still a bit of a newer thing.
Okay.
It kind of came about in like 2018, so it's still kind of a little bit newer. So maybe over time we'll be able to develop species richness into it.
You have
Right.
something more?
Can I ask a question?
Oh, you can ask a question, of course.
Thank you. I just in your permanent office, did you test the interaction between the time, the site, or because it's not clear to us that because you showed that NDS, so you did test the interaction part, right?
Yeah, between
Okay.
sites.
And another question, very general in a way. You said that species richness can provide functional redundancy. Do you think high species richness is sufficient to provide high functional redundancy?
Not necessarily, but I think it's interesting that it did have an effect.
And what do you need on top of species richness to like maybe have functional redundancy?
Probably some sort of like species adaptation, so their like ability to like prevail through high temperatures, also reproductive, like larvae settlement, should also be, they have to be high survivability of the larvae.
All right, thank you very much. We're gonna we started a bit late, so we are a bit over time, so we're gonna stop. Congratulations, Gianna. Congratulations for all of you that have presented this morning. We are going to take a short break, coffee break after hours. Not that short.
Not that short.
And we'll see. I need to move. When I'm doing ping pong, I'm going downstairs in the large room and realize. Yeah, I prefer this in the long run. To me, it's just like that. After half an hour, I start having pains in the lower
Okay, welcome everyone. With this we start the second session of the day. I'm Camilla Roveta, I will be the moderator. I'm from the Protection of Personal Market. We have 4 evaluators. Nicola. Okay, thank you. From Brest, UBO, Luana Monteiro from Ghent University, as you know, and Heidi Kampra and Katie O'Dwyer from ATU. And the first presentation is from Kylie with the title Quantifying the Vertical Distribution of Target and Bycatch Species in Brown Shrimp, Crango, Beam, Troll Fishes. The floor is yours, you have 15 minutes. When you have 2 minutes left, I will stand up. So you
Okay, thank you. Okay, welcome to my presentation on quantifying the vertical distribution of the target and bycatch species in brown shrimp beam trawl fisheries. So to start out, I'm going to give you a little bit of background on the brown shrimp fisheries in the North Sea. They are the most economically important coastal fisheries, supporting around 500 vessels, landing around 25,000 tons of shrimp annually. Now, brown shrimp are very small. They become marketable at only 5 centimeters, and they live within the seafloor, and so to fish them, the vessels use bottom trawls called beam trawls. Now, these trawls have a very fine mesh in the cod end here, which allows for the capture of these tiny shrimp, but allows minimal escape opportunities for non-target species. And so vessels are mandated to have bycatch reduction devices. The one currently used in the fishery is the sieve net here, which allows for smaller individuals to pass through to the back here, while larger ones move out of a bottom escape hatch. And while this allows for the reduction of these larger bycatch, it is insufficient at sorting out individuals of similar size to the brown shrimp. And so this leads to a persistent bycatch issue within the fishery, especially of juvenile individuals, as they fish on coastal nursery habitats. So why is bycatch an issue? Well, ecologically, a lot of time these individuals are thrown back at sea and they die, causing ecosystem-wide impacts and further depletion of stocks. But also economically for the fisheries, as more management is moving towards catch restricting policies, such as discard bans, under which the catch of other commercial fish species can cause the early closure of your fishery and economic losses. There's also a public demand for more sustainability certificates. The one which the brown shrimp fishery currently holds requires the ongoing reduction of bycatch within the fishery. So how do we work towards reducing this bycatch? Well, one way forward is understanding how species vertically distribute within a trawl to inform better bycatch reduction devices. So the first thing this can help us identify is if there's a natural separation within the species. So this other study here by Larson et al. Found that a lot of the fish were in this upper portion while the shrimp stayed low, and suggested a complete upper release of that 1/3 upper part of the trawl. It can also help us identify the placement of the devices and the escape hatches, as their effectiveness depends on species contacting them. So we can also see if maybe we would need a guiding panel to increase this contact and thus the effectiveness of the devices. And so this information is very important and can help us create better devices. However, it's never been looked at before for the brown shrimp fishery. And so my study aimed to do that, answering 4 questions. The first being, what is the vertical distribution of brown shrimp and commercial fish bycatch in the aft section, so the back section of a beam trawl? I further wanted to see if for the brown shrimp this depended on their length, and then if common gear modifications within the fishery, such as the sieve net which we saw, or a guiding panel, altered the distribution of the shrimp through the trawl. So to do this, I used data from experimental field trials that were held in the German Wadden Sea in the North Sea. These were done over 3 days using an experimental trawl in which the back part of the trawl was replaced first with this quad split frame, which vertically stratified it into 4 compartments. So we had the bottom, the mid-lower, the mid-upper, and the upper compartment. And as you can see here, this was placed at the back, so we're placing the cod end, and each compartment led to their own collection bag to keep the catch separate. To see how the different gear modifications altered that distribution, 3 different experimental setups were looked at. First, we had the baseline trawl with nothing impeding the movement of the individuals to the back. Next, we added on the sieve net here. And finally, a guiding panel was added after the sieve net. Each of these were deployed a total of 5 times, so 5 repetitions per setup, for a total of 15 hauls overall. Once the catch was brought back on board, it was kept separate per the compartment and sorted. For the bycatch, they were only counted and measured in the baseline trawl. And for the brown shrimp, subsamples from each haul were taken back to the lab, and photos were taken on a backlit board like so, with measurements. And I then placed these into image J and was able to get the individual lengths of each of the shrimp. So now for the statistical analysis part. So the main part of this was modeling that vertical distribution. Now this was only done for shrimp, as the bycatch was caught too sparsely across the hauls to allow for robust estimation. So for brown shrimp, the goal here was to estimate the probability that a shrimp at a given length would enter one of the 4 compartments. So to do this, we use multinomial logit models with length as our explanatory variable. Since no estimation procedure was available publicly within R, one was built using Template Model Builder. And finally, we ranked the models using AIC and BIC. If the length explanatory variable improved the fit of the model, we took that as evidence of length dependency. So for each of those gear setups, they were each modeled separately, but the 5 hauls within them were pooled together. So since we did that, we needed to account for that hall-to-hall variability, as well as we wanted to be able to make inferences based on confidence intervals. So to account for the uncertainty, double bootstrapping was used, and 95% confidence intervals were produced. The total output of this was predicted compartment probability curves with those confidence intervals, and these were then used to compare how those different gears altered the distribution of the shrimp. So with the 2 modified gears, we found the relative percent difference in the compartment proportions to that baseline trawl. Looking at the vertical distribution of bycatch, as well as we wanted to see if there was any difference in the proportion of marketable shrimp and undersized shrimp in any one compartment, we created the vertical distribution indicators, which is the percentage of catch in each compartment, with then 95% confidence intervals from bootstrapping. Okay, so on to the results. What did we find? Well, first off, 36,750 shrimp were measured in total, and length improved the fit of the model to the data in all of the gear setups. However, the vertical distribution indicators showed that there was no significant separation of the marketable and undersized shrimp in any one compartment or in any setup. So this is our plotted models here, starting out on the baseline. On the y-axis, we have the proportion of catch per compartment, and it's cut off a bit, but there's length down here on the x-axis, and we have it mirrored in panels with the bottom, the mid-lower, the mid-upper, and the upper compartments. The black line you see here is that estimated probability curve with the 95% confidence intervals, with the blue being the observed data. The vertical line here is that 50 millimeters where they become marketable, and the horizontal line here is 25%, so where we would expect these curves to sit if it was an even distribution. And what we see is that shrimp are predominantly caught in those bottom compartments of the trawl. So just this bottom half is around 75% of the catch across most of those length classes, with a lot less being caught in these top compartments than if it was an even distribution. Even just in the bottom, it's around 50 for most of the length classes. The trends we're seeing with the length improving the fit here is a slight increase in that proportion when you get a bit larger, but not huge differences between those undersized and marketable shrimp there. When we add the sieve net, there's an increase in the bottom with the line sitting around 75 for most of the length classes there, slightly larger confidence intervals on this one. But up top here, we have quite a large decrease in the proportion being caught No consistent length trends are seen here. It's a bit more of an S-curve on the wider ends of the length distributions. Adding the guiding panel, it's a very similar trend, again, around 75% of the proportion in that bottom compartment, and almost none being caught in those upper two compartments with very tight confidence intervals. The length trends here are a bit similar to that first baseline one. As you increase in length, there's a tiny bit of an increase in the proportion in the bottom compartments. So looking at the relative percent differences is what we have graphed here. So this is relative to the baseline. On the left is the effect of the sieve net and the sieve net and guiding panel. And again, it's by panels with the bottom, mid-lower, mid-upper, and upper. And on the y-axis is the relative percent change. And the horizontal line here is 0. So there's 0% of the proportion changed between the baseline and those additional gear elements. If the line sits below 0, it means that the proportion decreased when you added the sieve net or the guiding panel. And if the line sits above the 0, it means that the proportion in that place increased. If the confidence intervals are in green, it's significant. So what we're seeing generally is that the distributions are shifting downwards across most length classes when you add those gear elements. So for the bycatch, there was 4 commercial fish species or species groups analyzed. This was the common dab, the European plaice, sand eel species, and whiting. So this is our vertical distribution indicators. On the bottom here, we have the bottom compartment, mid-lower, mid-upper, and upper, and the percentage of catch in that compartment on the y-axis, with the horizontal line going back to that theoretical vertical even distribution. And what we're seeing is that they're predominantly caught in the upper compartments. So the point estimates here are the numbers with the confidence intervals. Common dab around 45% in just that upper compartment. The European plaice around 45 in the mid-upper. Sand eels, we have about 50 in the upper, but the confidence intervals are a bit more uncertain, so it could be closer to an even distribution, with whiting having quite a difference of 57.4 in just that upper compartment. So, how do our results fit into what we know about the vertical distribution of species within trawls? For the brown shrimp being caught lower in the trawls, this aligns quite well with what is previously known with other crustaceans and shrimps. And their movement is thought to be passive through trawls. So if they're caught within the sediments, they're brought into the trawl, they'll stay low, tumble through to the back. There was some caught in those upper compartments, however. This could be due to some pre-capture positioning higher in the water column. However, inferences can't be made strongly about that since our stratification was only in the back. With the downward movement with the addition of the sieve net, this opens up the possibility of adding these secondary size sorting grids, which have been looked at and studied in other shrimp fisheries, in which it has a bottom escape opening, allowing for undersized individuals to escape out, while larger ones are brought over the top and then into the cod end. Since the sieve net moves most of that catch already down into the bottom region of the trawl, most of the individuals would encounter this and be able to have a secondary chance to leave and be left in the sea. For the bycatch being caught predominantly higher in the trawl, with whiting this aligns quite well with previous studies. No studies have been found for sand eels, so this is presumably the first. For common dab and plaice, this actually contrasts previous studies, which found them lower, which is what you might expect for bottom-dwelling species. However, comparison is inherently difficult because any operational, technical, environmental conditions can change the species' interactions with the trawls and thus their vertical distribution. All of the compared studies used otter trawls, which have large nets out the side, which could alter their interactions and distributions. So what are the implications for bycatch reduction devices? Mainly what we found is that an upper escape would offer a more natural escape route for the fish compared to that bottom escape of the sieve net. This would offer less time in contact with the selection device, so more chances to escape and not pass through to the cod end. One way we could do this is through sorting grids. So those are these grids that you're seeing at the bottom here, which work in the same size-based fashion of smaller individuals passing through. These are completely legal within the shrimp fishery, however, are underutilized due to the lack of knowledge and research on them so far. Looking at the natural separation and if this would be applicable to this fishery, although there was that separation in the individuals, the loss of marketable shrimp would still be unacceptable likely. You have around 25% in that upper half of the trawl. So if you were to split it, you're losing a lot of marketable shrimp. Fishermen are probably not going to like that too much. There is the possibility to add stimulus devices in net prop fisheries. They have found that these have successfully driven a lot of fish species into those upper portions of the trawl. However, for the shrimp fishery, if this would to be looked at, they should simultaneously see if they can move the shrimp downwards while also moving the fish upwards. This study did come with some limitations, mainly that it was a short study period, so 3 days, which is not reflective of the full fishing conditions of this fishery, which is year-round and at night. And shrimp and fish behavior is known to change with environmental and operational different conditions, such as light, temperature, tides. And so the results of this study and the implications of these devices need to be tested across a wide range of operational conditions to allow for robust devices to be created. So overall, what this study found is that brown shrimp are predominantly caught lower in the trawl, while the fish bycatch is caught upper in the trawl. So what we know is that better bycatch reduction devices are possible. This study suggests that upper escape sorting grids and the development of strategies that utilize natural separation could be a way forward to allow us to reduce bycatch within this fishery. These are my references, and thank you.
Thank you, Kaylee.
Thanks very much, really nice presentation, and of course I have to comment on your matching hand knit.
Thank you.
A theme of what I've seen in your workshop over the master. So really nice presentation. Actually, in the discussion you already you preempted some of the questions really well, so there was a few questions that you've already answered for me about the grid size, that panel that can be added. And then one that you commented on, and I was going to ask when you were talking about the fish stimulus, was about the shrimp stimulus. But I'm wondering, what are the proposals or what are the developments that are ongoing in trying to do that, trying to separate the fish and the shrimp? What sort of devices or what attractants are they using for that?
Yeah, so one of the main ones, back here. So for the shrimp and the other species, to increase that contact, they do a lot of times use guiding panels. So they move everything down, but in theory that moves everything down, so maybe you wouldn't want that for the fish bycatch. So the simultaneous movement of both species in opposite directions, I haven't read anything that anyone's tried to do that. All of the studies of these stimulus devices come from net prop fisheries, which they generally find them lower in the trawl, so it's not much of an issue to get the net props down. So these bars have been proposed a lot. The fish hit them and move up. This study here, actually by Amelia et al., they also looked at chains, putting chains over the bottom, and rising floats. So they put like buoys and floats that move like this, and so in theory, hopefully it just slowly guides the fish to move upwards. I have seen some with lights as well. They try like blacking out the netting in certain parts and putting up LED lights. The ones that I read weren't super successful. Even these ones, in theory, they get successful results and it moves up, but having results that are successful within fisheries and implementable, that the fishermen find that there's enough of a change that it's going to actually really help, aren't to fruition just yet. Okay. So it's quite a new area that people are still working to create that separation more successfully.
Yeah, okay, interesting. And then my second question was kind of on sustainable fisheries and the future of sustainable fisheries. Do you think, or have you come across, like, should we be trying to improve trawling for the species, or should we be using pots or something different?
Great question. Yeah, I think the ideal is moving to more static fisheries, and that's actually another big kind of working question within my working group that they're on right now. So one question is if it's possible with pots. There's a full project going on with that, and there's just so many different elements to it, and they're really trying to design like what type of pot, bait, lights, and if it's going to catch enough, because these shrimp are so small that it's also really hard to catch enough to make it economical for these fishermen to want to shift from something where they can just sweep the seafloor to where they can put one pot down and it collects enough shrimp. Another option that they're also looking at is stow nets. So same kind of thing, like it's a static net. So kind of like a trawl, but it just sits in the water column. So you're not dragging it, but it's still catching everything that gets pushed into it. And that's also shifting with our understanding of shrimp ecology. So it's thought that usually they're staying on the seafloor, but more and more they're finding that they are at times coming up into the water column. So that's another option forward. But yeah, definitely a shift would be more ideal as we progress and fuel gets more expensive and it's, yeah.
Okay, that's it for me. Thanks very much.
Thanks very much, Kylie. I thought your presentation was excellent. I think your work is extremely important, especially with the discards ban, so this is very, very needed. So I understand that obviously you had limited time to do your sampling, but if you had more time, what would you change in your experimental setup and how much would you change that?
I think for the fish species, it would be very interesting if we had data on all of the 3 setups, because that could also answer the question if they're moving downward when you add those sieve nets or those guiding panels, and then if you could then again move them back up, because it's possible if you have the guiding panel, they're still moving back up, and that could answer our question there. Adding more sample sizes would always be ideal to allow for the modeling of the fish bycatch. Here would be, yeah, definitely ideal. And then I think if I'm moving forward with this understanding is trying to work towards more of these like escape panels or the better bycatch reduction devices. Another thing I definitely would have changed, with the more sampling question, is that short study period. So one of the major things you could have done even within the short period is fishing at night, because as I said, they think that brown shrimp move at certain times, and one of those is at the nighttime to move up and feed within the water column. So if you're finding that they're moving passively through the trawls, getting caught in those higher parts, if you have an upper escape, maybe at night you're losing 50% of your shrimp. So I think that's already one step forward that could be easy to implement into the next study.
Then why did you not look at the fish species as well, the bycatch, in
Yeah,
all of
so-
your
Yeah.
treatments?
First, I was given the data of the experiment, so I wasn't unfortunately there for the setup. So why the data wasn't there is a little bit, not understood, but it would be very interesting to have it, to be able to like fully compare between the different setups.
Are you going to continue on this work? Or?
Yeah, hopefully be able to put this forward and also publish within this. Yeah. Thank you.
Kylie, congratulations. Excellent presentation. As for questions, I had a similar question as Heidi, and I was going to ask you to elaborate more on future studies to confirm findings. So, but you kind of already gave a lot of indication of that. But if there's something that you would like to add more, that from what you have been explaining to us, besides the behavior of the shrimps, but and increasing sampling time. Did you have a chance to give a thought about the outreach, or what would be in an ideal setting, let's say, if it would be easier to reach out and establish a study? Do you have a thought about environments, based on locations where the species, the market, that is sampled? Did you have a chance to think about that? What would you like to do to really further improve the significance, not only of our results, but if they are confirming also to expand it?
I think I understand what you mean. If you can get more robust results, so if these kind of things are brought forward. And then I think the next step, especially within fisheries, is finding devices in which the fishermen are willing to move towards, and you're able to communicate with them and kind of like bring it into their trawls easily. It has to be something that's implementable, that they can shift their gear towards. And all of this was done just within like the Wadden Sea, which is where, all along the north coast is the most of the shrimp fisheries, and then they also have them in the UK as well. So also, I guess, collaborating with different scientists and seeing if in different waters these are similar, because sediment type is also known to shift the distributions. And so if this works in the German one, see with German fisheries, it might not work in the UK if they have slightly different sediment types and the plumes are different. And so yeah, I think just generally across every different environmental condition you can think of, yeah.
But also, would you, if you have the chance to put time further, do you think it's important to also document, as you're saying, all these kind of considerations of sediment, environmental conditions, and so on, to stimulate other scientists to and policymakers to implement this study in other relevant regions?
Yeah, I guess that is something that would be very interesting, to add these environmental factors as explanatory variables if you're looking at what is shifting it. So being able to say, like, if your sediment is this type, if you fish at night, and if you do this, this device will work better. Or if you're fishing in these conditions and your sea is this temperature in the summer, you should use this device. So yeah, I think implementing and being able to share that across different regions and not just this one area.
Yeah, thank you, Gary. Really liked your talk, very clear, I think. But I must say, you lost me a little bit with the results at some point, and I would like to come back to your graphs.
Yeah.
Because I've the first ones. Yeah, before that.
This one?
No.
Like very first ones. Yeah.
Yes, those ones. I did not understood what are the points in those graphs. They are like individual shrinks, or
They're the observed, so they're the pooled of the observed. So if we take one point, for example, like shrimp at 20 centimeters, so it's length classes. So we class, so if you have like 100 shrimp that are 20 meters, like they're here, and you're going to find about 80% of shrimp at 20 centimeters here. Or, 20 millimeters, sorry.
you mean 80% in between the different?
So all of these are correlated. So if I have if I have like a 20 millimeter shrimp here, and 80% is here, and 10% is here, 5 or something along those lines. So they're a pooled class of how many at that length.
And so then you have only 1 point per size class?
Per size class, yeah, exactly.
Cool. I guess I was a bit lost. Like, how can you have a proportion and a size at the same time? But that makes sense.
Yeah.
Then I'm wondering. I don't know. It's very. I would have approached the problem a different way, but it doesn't mean that the way you did it is not right. And do you have an idea of what that would look like? And have you looked at that in terms of size spectra that you get in the different groups? And would that look like exactly the same, or
like how many? Like
with
if you have
basically like an histogram of the different size population.
I have that. It's not in here. I didn't add it to my extra slides, but it's in the thesis. Like beside each one, you have the histogram of the shrimp length classes. And they're generally like a pretty normal curve. And that's also why, like, if we look at any of them, you have wider confidence intervals usually at these lower ends, because most of the catch is within this middle compartment of that general histogram. So our estimates usually in those catches where we have more sample sizes, the curves or the 95% confidence intervals are quite a lot smaller, and then they kind of fan out. And I think it's also here you can see a lot, like here there might have been only 10 shrimp that were 80 because it's more rare. So it definitely does affect, and that's where we're able to use then these confidence intervals to make our inferences.
Yeah, so this pretty shows like you have like an hard time in a way. I mean, not you, like the net has a hard time separating the different size classes of the fish. Just like they pretty much all go in the bottom. Nice. I had another much more general question, I guess, because I'm not a fishery guy. And when I saw like all these big devices and everything, I'm always wondering about like how the one you managed to either fish or shrimp that you managed to get out of the net. How are they after that? Like, do you have an idea of do they survive? Are they injured or?
Like when they, if they, oh my gosh. Like if they're able, when they go through with the escape.
Yeah, the one that go out.
Yeah, I haven't seen any studies on their post-release of just like the seed nets. I think that would be very hard to study, especially in these tiny shrimp.
Yeah.
Being able to see if they move through. I do know there was a study in 2022 that looked at their post-release survival after being brought on board and the undersize being released, and it was not very good. However, shrimp are very handled when they're brought on board. The way that they move them out is through these giant sieves that are like tumbling them around until they're brought out. So it's not understood if just going through the trawl and being moved out would really impact their survival. But it would be very interesting to know that.
Okay. Do you have an, I don't know if we have time.
1 minute.
Do you have any idea of how you could do that?
I think
I don't know.
the best way I could think is like trying to simulate it the best you could in a lab. You could attempt to have a secondary device, like if you had the escape and you have like a lot of times within fisheries they have like secondary nets that catch what escapes, and that's kind of how you can compare retention rates. But then you're putting through another stress of being brought up to the air. So really isolating just the effect of that one movement, I think would be very difficult. They do have flow tanks, I guess would be another option. So where they test actual gear within these giant simulation tanks, and you can see how the gear moves through. So that could be an option to simulate it if you had shrimp within that and you kind of push them through and see how they do. But again, in any study where you're also taking an individual out of its environment, there's so many factors that then you have to fish it in the first place to bring it onto land to test it, and lots of factors. So it'd be very, very difficult. Yeah.
Thank you.
Yeah.
Thank you, Katie.
Thank you.
Congratulations again. See? The thesis is CO2 emissions in the canned tuna market, insights from the Spanish versus CN fleet and international transport. The floor is yours. Again, after 13 minutes, I will stand up, saying that you have 2 minutes left.
Okay, so good morning everybody. I'm C, and I'm happy to be here to present to you guys my thesis named CO2 emissions in the canned tuna market. Insights from the Spanish Persian fleet and the international transport. So, nowadays the increasing anthropogenic CO2 emissions has caused a big environmental pressure to our planet. And how to get a better understanding of it and then to reduce the emissions is a task that we're facing in common. And it's important work frame inside the United Nations development goals. And in these days, the fisheries are increasingly recognized as an important fuel consumers, generating a lot of CO2 emissions. And at the same time, the globalizing seafood market also increased CO2 emissions related to the transport and logistics behind the trade. And within this context, the tuna species are among one of the most exploited and traded marine fish. And among all the tuna products, canned tuna plays a particularly important role here. And they are mainly made by tropical tuna species, and they are skipjack, yellowfin tuna, and big-eye tuna. They are mainly harvested by the tropical tuna Persian fleet fishery. And beyond the fishery stage, the trade of the produced canned tuna also relied on very international and distant multimodal transport. So therefore, our study decided to investigate the CO2 emissions related to the fishery stage and the international distribution stages of the canned tuna market, one of the most important seafood market in the world. And so our specific objectives is to quantify the CO2 emissions associated with tropical tuna person fleet and the international distribution of canned tuna. And for both objectives, the emissions are quantified in absolute terms and also in relation to the volume of the products. And here, specifically, Spain is chosen as a case study due to its well-known leading role in the global canned tuna market. And in order to offer a current overview of the CO2 emission status, we use the latest dataset and covering a study period from 2022 to 2024. And we use the vessel monitoring system data and vessel-specific characteristics of Spanish tropical tuna person fleet from the Spanish government. And the VMS data here is mandatory for fleets that is longer than 50 meters in Europe Union to report their position, velocity, and time every 1 or 2 hours. And the fisheries dependent information also from European Union is used for the tropical species landing data. And the canned tuna export data is from the UN country database under this category. So the CO2 emissions associated with the fishery is a function of the engine power, the CO2 emission factor, and the load factor of the engine, and the time interval of the VMS data record. And also the fleet velocity of, in the VMS data are used to calculate the load factor of the engine. After that, the emission divided by the landing of the tropical species are calculated as the fisheries emission intensity. And for the international distribution part, similarly, the CO2 emissions is quantified by a specific, a transport mode specific CO2 emission factor multiplied by the distance of the road of the export flow and the mass of exported canned tuna. And similarly, the emission intensity for each transport mode are calculated by emission divided by the volume of canned tuna. And finally, for this stage, we adapted a network analysis and to visualize the distribution of the emission of the transport emission. And we use the nodes and edges to represent the export partner countries or territories, and the export flows connect these export partners with Spain, respectively. And the strengths of the nodes and edges represents their transport emission importance. Okay, so first with this map showing the trajectory of Spanish tropical person fleet in the whole period, we calculated the total emission with an annual average, and the vessel average emission, and also the total fishery emission intensity for them. But if we zoom in to see the result in the 3 oceans, we can see that Indian Ocean is definitely a hotspot, and with highest share of the total emission and the largest and the biggest number of vessels, followed by those in Atlantic Ocean and Pacific Ocean. However, for the fishery emission intensity, Pacific Ocean has the lowest number, which means they are the most emission efficient ones. And this kind of various in spatial results is related to also the fishery strategies they choose to use. For example, here we have free swimming school and fish aggregating devices could be used. And compared to FSC, FAS is decreased the searching time for fish and also increased the catch success rate. However, it's more fuel intensity regarding the every unit of the volume of landing. And for the international distribution part, similarly, we calculated the total emission with the annual average and the total transport emission intensity regardless the transport modes and the number of export partners of span. And in this study, the emission in fishery stage is much more than the emissions in the international distribution stage. However, combine them both, the emission volume is still very high and it's higher than those emitted by 32,000 cars in the same time period. Besides that, the fishery emission intensity here is higher than the transport emission intensity, and this result is consistent with other researches also. And if we also move into the result in the different modes of transport, we can see that for the air mode of transport, they have the highest emission intensity, while the road transport accounts for the biggest share of the total emission, and the maritime transport connected the most is the most internationally connected. And for the network, here we can see that with the highest emission intensity, the air transport mode connected the fewest export partners. And besides the most important trade partner, United States, the strengths of the other export flows remain almost the minimum. And for the road transport, accounts for the highest share in the total emission, but the network is limited only this area. And here we can clearly see that the road transport is mainly used in export flows towards Europe export partners. And additionally, the fact that Spain export most of their canned tuna to European Union countries also related to the the importance of the road transport we found here in the emission. And for the maritime transport with the lowest emission intensity, it's the most internationally connected. And here we can see the strengths of the export flows are much more even, and the network is more well spread. With the Great Britain being the most important export partner, the maritime transport still holds higher total emission compared to the air transport mode. However, the air transport mode have the highest emission intensity. And for the future study directions, I have to say the formula we use to quantify the CO2 emissions in fishery stages is adapted from universal methods which means it's useful for many kinds of ships. So I think in the future, a tropical tuna perceiver specific method or function to quantify this is essential. And also we can adapt some detailed operational data to have a more accurate estimation regarding the different fishing activities, such as fishing, searching, and steaming. For the international distribution part, out of the limitation of the dataset we choose to use, the UN country dataset, we didn't consider the middle stop and the change of transport modes here. So in future study, we hope the shipment level logistics data could be involved to reduce the uncertainty in transport related emissions by fill these blanks. Okay, so the conclusion of this study could be the volume of the CO2 emissions in both sectors in Spain is high. However, if we can consider all the countries and all the sectors in the global canned tuna market, this result will be even higher. And the fleet emission efficiency is related with the operating ocean and the choice of the fishing strategy used. The international distribution emissions and its emission efficiency is the balancing interaction of the mode of transport and the geographically distribution of the export flows. And here I want to say thank you to my supervisors and the Greco research group in IU, and also my friends and my fellow students and my family.
I'm first.
to start?
Oh,
Yes.
Thank you for your presentation. I don't know, I had a question, but you managed to answer it in the perspective, so I guess that's good. I had a more technical question on the first indices you used. If you can go back to like before.
Like the message, right?
Hmm?
Sorry.
A question about the.
Oh, okay. Yes.
So the indices you use for the slide where you explain your equations.
Yeah.
So this one for the CO2 emission associated with the international distribution.
Yes.
So the M on the equation, it's already the tuna mass, right?
Yes, the mass.
And then you remove it to get your emission intensity for each transport mode, right? Or is it another method that you use? I was a bit surprised to see that the mass was in there, and then you control for it.
Okay, so this one I use it because I calculated for different modes of transport.
Yeah.
Then for every mode of transport, they are used for to transport the tuna to different export partners.
Yep.
So they have like different partners. They have different distance in this equation.
Yeah.
Yeah, so that's why. So it will not like remove it. Will be simply the.
Yes, so that's it. Yeah. It's the same. That was the thing. Like the M in there is just the same thing as what you divide by.
Yes.
Yes. Okay.
The mass.
Okay. That's it.
Okay. Congratulations. Presentation really good, really nice slides, and very clear, relevant work. Could you show the slide where you show the road emissions, where you focus on your results, that's concentrated in Europe? You know?
Okay.
Yeah. have you had a chance to reflect on it based on the results of your study to provide any kind of advice to policymakers
Yes.
in the European Union of what could be done to reduce the CO2 emissions for tuna transportation on land within the same continent?
Okay, so okay, so you mean just for the transport modes?
Yeah, so you're showing with your study 97% of
Yes.
the emissions are due to the on-land road transportation and further
Yes,
because it's within actually the same continent. Did you have a chance to think which other alternatives could be to lower
Okay, so
this?
well, the thing I thought is if they could use more the maritime transport, the emission will be much better. I mean, because the emission intensity here you can see it's really low. It's 0.07. However, for the road is it's like 3 times higher. Although it's still lower compared to the air transport mode.
Well, I'm not just to continue the conversation, so because I'm curious about it. Do you have information about the points where what it happens on land? I was just wondering how far this alternative could work throughout the countries that are receiving the tuna by road transportation, because there are certain limitations with the maritime transportation inland. So you want to see what I mean? You could reach the harbor, but I guess you'd still need to use road transportation.
Yes.
And maybe not to every
That's also a
country.
question. No, sorry, not also a question. It's like this also important. It's like we cannot just tell everybody to use the most efficient thing, the way to do this transportation or fisheries, because it's like related to also not only the environment but also the society. It's like so I feel the most important thing, the core suggestion I can give for the stakeholders will be how to balance this. How to balance that one in one hand we reach the how do you say that the demanding of the market, and another hand use the most efficient ones we can use.
Yeah.
So I feel
Yeah,
that's
I agree with
answer.
you that is key, because then maybe a combination of train transport as well, I guess. But yeah, it all comes to the understanding
Yes,
of
like
that
for the
of the
air
need for
transport modes,
balancing.
it's like, well, if the canned tuna is produced and it's shelf stable, it don't need to transport really fast. So why still there so many people, so many export partners choose this transport mode? It's like because they can transport it directly, and if it's by ship, you have to after the port you have to drive. So it still exists, although it's very not eco-friendly.
Yeah, okay. I'm satisfied. Give the chance for my colleagues to ask questions. Congratulations.
Thanks for your presentation. I may have missed, but how did you collect the information from the export flow?
Okay. So I use this data set to get the information about the like the total export of span in the canned tuna area, and I got it's like a list of the export partners. And however, for the road of the export flows, for maritime ones, I use a special data set for the I don't know, it's like from a country to another country, the official sea road they use. I access that data set. And for the road data set, I use like it's from an R package to calculate. It's like related to the map, like a Google map, and to calculate the distance from the from the two countries. If we have their I can show you this. If we have this the latitude and longitude information, then we can calculate the distance.
And then, do you think that, I mean, you mentioned that you also got your fisheries data from the vessel monitoring systems. Do you think you might be missing a lot of data on that as well, from like illegal fishing and that kind of stuff?
It's true.
Yeah. And do you think that would change a lot your kind of results?
But actually, like this kind of tuna fishery is very industrial. So it's like it's also one of the limitation is that we only consider the CO2 emission from the fleets, the vessels, the main vessels, but we didn't consider the CO2 emissions used by the feds and also the support vessels. Like we didn't consider them.
And lastly, you mentioned that in the Pacific they were more efficient in terms that they were using the fish aggregating devices. Do you think that's a type of advice that like policy could make for other types of oceans? Or?
Yes, it's like this is one thing they can do is to regulate the choice or the use of the FSC and FES because. It's although I mentioned like FICE here is more fuel intensity, but however it's also showing out the research is like the FSC because have a longer time for research for the search for the fish schools, so maybe they can also cause sometimes they can also cause bigger CO2 emissions, so it's a very tricky balance.
Okay, thank you.
Thanks very much. Really nice presentation. Very interesting study and question. I was wondering, so kind of related to some of Heidi's questions, but about the data. You know, obviously you get data from these databases, but it's not always in a format that you can use and everything. I was wondering if you can mention what was involved in the data cleaning and preparation before you could even start your analysis. So when you go to the VMS database, when you go to the Comrade database that you had, I guess you had a lot of data organizing, cleaning, restructuring.
Yes.
Yeah.
You want me to explain?
Yeah.
Okay.
If you can expand a bit on just like the time involved or
Yes.
how much of an issue was it to get it ready for your study?
Okay, so first is I removed the logically overlapped data. It's like in one time a vessel cannot be show up in both two points. So from the I clean the logistic overlap, and also I clean those have really high velocity that they cannot reach, and also the time interval is I need to select like from for a time threshold that is reasonable. Because for example, if it's just the two records, it's 24 hours difference, and it just means it didn't work.
Yeah. Okay. Yeah. And did that take long? Like organizing all the data? How much of the time? You know, because as soon as you get to analysis, you know roughly what you're doing. But I'm curious how long this took.
Oh my god, my brain.
No worries. I was just wondering like how big of a task it was, but don't worry, that's fine. I have a couple of other questions. So I thought how you present the information is really interesting. Like your focus is clearly on emissions, but of course all of this costs money and the world runs on money,
Yes.
unfortunately.
Yes.
So I was wondering, how does this relate to the costs? Like, is the air transport more cost efficient, so they want to use it, or does it get them to a market where they can get more money for the tuna that they sell in that market, or, you know, what's the reason to keep certain modes of transport related to cost?
Yes.
And I don't think you have the cost data, but I'm wondering if you can just comment on it,
Mhm.
or if you've seen other studies.
Yeah, and as you said, the air transport mode is it costs higher for the financially. And
Costs higher.
yeah, you can, like, for example, like you can see yeah, it's because when I doing the research, I search the exporters, the developing country or the developed country,
Okay.
like among the list.
Yeah.
So here you can see like the Latin America and in Africa, the network is limited. And but for the sea mode transport, because it's not only the most efficient for the emission, but also it's a cost emission. Emission cost. So, like, it's used by the most of the export partners, and you can see it's very well spread.
Yeah, but my question is more like, if I'm a Spanish tuna production facility
Okay.
and I want to sell my tuna for the best price, am I going to fly it to the US, or am I going to send it on a boat to the US? Or, you know, that's more my question. So it's like redoing this study, but instead of looking at emissions, looking at cost. I was wondering if you came across anything in the literature on this, the economics of this whole enterprise.
Well, it's a very conductive question,
Yeah,
but
yeah.
right now I'm sorry. maybe I
It's
cannot
just
answer.
on the emissions. Yeah, that's fair. Yeah, I think we need to get to that. You know, joining the emissions with the economics to actually implement changes. So I was just a bit curious.
Yeah.
in there. And then the other question I have, is there 1 minute? Or?
Yeah.
Yeah. So you, okay, very quickly. You compared the emissions at one point to 32,000 cars, but for me this is like a pie in the sky. I don't know what 32,000 cars means. I was wondering if you came across the emissions more in comparison to like tamed sardines or beef production or some other thing that's like a similar nutritional value for the human population, you know, some relative comparison to tinned tuna emissions rather than 32,000 cars. Could you compare that to some other food production?
Okay, so I try to present
Yeah.
to compare it and
Yeah.
written it in my thesis.
Mm.
But it's like for the things you mentioned, like
Yeah.
beef or chicken, whatever.
Mm.
They are more like for farm.
Yeah.
And for them, the most pollutant air is not the CO2.
Emission.
Yeah,
Yeah.
so they have other emission like
Yeah,
CO or whatever.
Yeah.
So they usually in these papers they use a different unit than what I use.
Okay,
They
so
use
you
the
can't-
CO2 equivalent.
Okay.
So yeah, I didn't put
And
it
it
here.
hasn't, this hasn't been done for like sardines or mackerel or something for comparing.
Well, it's like it's very limited in this area. So
Yeah.
it's like
Yeah.
what I compared now is
Yeah.
already use some information from the other canned seafood and also other frozen seafood or fresh
Okay.
seafood.
Yeah. Okay, thanks very much. Well done.
Thank you, Si. Okay, now on this side. With the thesis, space and time patterns of the mixed species in response to multifaceted climate change metrics across the Mediterranean. Okay,
perfect. Thank you very much. I know it's a mouthful, the title.
Sorry.
But yeah, thank you all for joining me here on my master thesis defense. My name is Louise Constance, and I'm gonna repeat it one more time. So yeah, the space and time patterns of demersal species in response to multifaceted climate change metrics across the Mediterranean Sea. Well, it basically comes down to the core question. If the Mediterranean Sea warms, what do the species do that live right on top of the sea bottom? Do they move somewhere, and what are the specific drivers that are moving them there? So why does this matter? Well, I think we're all quite familiar with climate change and the effects of it. More than 90% of the excess heat generated by human greenhouse gas emission have already been absorbed by the oceans, and because of this, this has like a big effect on everything that is living in there. And specifically in the Mediterranean Sea, that has been ranked as one of the most vulnerable climate hotspots globally. In the North and Barents Sea, we already have like results that 20% of the demersal fishes have been showing detectable shifts to other regions. But there is a big research gap because no prior basin-wide studies have been done for multiple species and multiple climate metrics simultaneously. The expectations here are, I'm not sure if you're familiar with the meridionalization hypothesis, which basically means that species are tending to move more towards polewards regions and also towards deeper waters because they are trying to escape the heat. So these expectations have been challenged by research observations in the Mediterranean. So because of this, my core hypothesis has been a bit adapted. So I think that redistribution patterns will vary geographically across the basin with stronger and more consistent shifts in regions that experience more intense climate change effects. My two main research objectives are, the first one is to characterize the extent and direction to which species move, both in latitude, longitude, and depth. So are they moving towards more northern waters, east, west, deeper or shallower? And then the second research objective is to relate these observed redistributions with climate drivers that I will explain in more detail in a minute. So I work with the MEDITS survey data. The MEDITS stand for Mediterranean International Bottom Trawling Survey. It's been going on for a couple of years, but I work specifically with data from 1999 till 2021, so 23 years of data. The surveys were done annually between May and July, and we almost have 26,000 holes. A hole represents when they put the net down at a specific depth, and also at a specific timing, and in one of the Mediterranean subregions. I have 30 in total, and you can compare them to like ecoregions. So each of the 30 ecoregions represents a similar species composition. The depths were between 51 and 800 meters, and all demersal species were catched in those nets. So fish, elasmobranchs, crustaceans, cephalopods. And then on this map, you can see the sampling locations of all the holes. The blue dots represent all the shelf habitats. So these are the habitats below 200 meters. And then the orange ones represent the slope habitats, which is more than 200 meters of depth. After gathering all the data, I had to put certain filters to only work with sufficient species that were present everywhere in each of the subregions, but also over a certain amount of time. So I had a temporal filter, a spatial one, and an abundance one. I started with more than 1,400 different species and ended up with the 250 most abundant species. After the filtering system, I wanted to know the geographical location of each and every one of those species within each year and within each subregion. So I used a spatial indicator, which is called the center of gravity. It's basically the abundance weighted mean of the population. And then you have information about where the species is located in latitude, longitude, and depth. These were the calculations that were done. And on the graphs, you can see that I created a temporal trend for latitude, longitude, and depth. For example, here you have a positive slope in latitude, which means that the species are moving towards more northern waters. For longitude, if it has a positive slope, they go more towards the west. And if it's a positive slope in depth, they move towards deeper waters. This spatial indicator has been frequently used in other studies as well, for example from Thesis and from Sans Martin. And then these first three questions were where to answer the first research question, where do the species move to? And then the next two is to put the environmental layers on top of them and see if there's a relationship between the distributions and between the different climate metrics that I used. So I had 7 climate metrics in total and 2 geographical metrics to put into the model to have a better look at the spatial distance in the model itself. So here you have a look at all the maps I created. The climate metrics I divided into 3 different dimensions. So we have acute, which represents the intense marine heat waves. Chronic are the long-lasting warming trends, and then seasonality represents spring advancement and autumn delay. And then the last part, of course, is the statistical part. I created linear models for each of the response variables, which was the trend in latitude, the trend in longitude, and the trend in depth. All of the predictors combinations were used, but before doing that, I had to check for collinearity and got rid of 2 of them because they were, too multi-collinear. So the marine heat wave mean duration and the center of gravity and longitudes were not remained in the models. Then the best model was kept for each of the response variables, and I created partial residual plots to have a look at the climate metrics and if they have if they can show any geographical pattern based on the redistribution of the species. So a quick reminder, the first objective was to see where the species are moving to. And I discovered that it's a basin-wide phenomenon. So in all of the 30 Mediterranean subregions, significant shifts have been shown. Almost 1,900 significant species subregions observations were detected of the 250 universal species. 66 of these species, which represent 30% of the total amount of species, showed significant shifts in all 3 dimensions, so both in latitude, longitude, and depth. And in this table, you can see I divided the Mediterranean into West, Central, Adriatic, and Eastern. And if you have a look at the numbers, you see those are representing the significant shifts, and they are decreasing while you're moving towards more eastern subregions. One ecological explanation of this is because the Western Mediterranean is in a more active phase of redistribution compared to the Eastern Mediterranean, which has been more exposed to climate change effects already. Then for the horizontal redistribution patterns, these are specifically the latitudinal shifts, or north or south. Here it was important to see that the subregions could be divided into north and south of the 40-degree north parallel. So subregions that were located above this line were tending to move more towards northern waters, below this line towards more southern regions. But there is no uniform poleward shift like originally expected. Then for the longitudinal shifts, you can see that in most subregions they are moving towards the east. The westward shifts were less prevalent and more localized, but in literature this has not really been studied yet, but it would be an interesting axis to also take into consideration. Then for the depth shifts, also here like the latitudinal shifts, we don't really see a uniform direction. The shallowing trends were taking more place in the Western Mediterranean, and then the deepening trends in the Eastern Mediterranean, but also in the Adriatic Sea. We also see that the magnitude is less compared to the horizontal distributions. Then for the second research objective, we're putting the climate drivers on top of the redistribution data to see if there's a relationship there. So first of all, this is the latitude model. Here we can see the 3 climate metrics and the geographical one that we retained. We have a look that all of 3 of the climate metrics were retained in the model, and the more intense these climate metrics became, the more southward species tended to move. And on the right side of the dia, you can see that I made partial residual plots. These are quite scary to look at, so I'm quickly gonna go through it. So basically all the colors that are darker presenting the Western Mediterranean subregions, the lighter color are the Eastern Mediterranean ones. And then each one of these horizontal lines are representing a subregion. And each dot is one specific significant result detected in those subregions. This particular plot is the mean spring sea shift, which means that you can see like a gradual change between the Western Mediterranean and Eastern Mediterranean. So the Eastern Mediterranean subregions are experiencing a more intense shift in spring advancement, which means that those countries, compared to 10 years ago, spring is coming earlier than expected. And this will have a big effect on the direction as well of the demersal species. So for the longitude model, also in this model, all three of the climate metrics were retained, as well as the geographical one. And here we have a look at the partial residual plots. In comparison with the previous one, you don't really see a pattern going on, a geographical pattern, because all the colors are quite intermixed. Then for the depth model, it is important to notice that the acute climate metric was not retained, which means that marine heat waves don't really have a big importance on the shift in depth. And another interesting thing to see is that both the seasonal metrics were retained, and they are contrasting because the spring seasonal shifts, they tend to go towards more deeper waters, and then for the autumn seasonal delay, they are tending to go towards more shallower waters. Then for the partial residual plot, you can see that the temporal trend doesn't really have a gradual increase or decrease, but it's more split into two. So the western subregions and the eastern subregions are on opposing sides of each other. So the discussion, what does this really mean? Well, first of all, the meridionalization hypothesis, which means that more polewards and eastwards shifts, is only partially supported. Then secondly, the seasonal asymmetry is a novel insight. As you might remember from the depth model, the spring seasonal shift and the autumn seasonal delay had contrasting results for the species distribution. Then third of all, the marine heat wave intensity drives horizontal movements, but is not incorporated into the depth model. And then fourth, the chronic drivers, so also contrasting mechanisms. This is for the longitude model, where we saw that for the temporal trends, the species were tending to move more towards eastern subregions, and then for the climate velocity, more towards the west. I faced a few limitations in this study, and the main one was that for the calculations of my climate metrics, I only used sea surface temperature. So in the future, I would like to include bottom temperature as well, because we are working with demersal species, and this would be really interesting to look at. But also vertical climate velocity, which basically means where in the water column species have to move to in order to remain in the same thermal conditions as they were previously in. Second limitation is that for the model making, we had to look at multiple species at the same time. And because of this, species-specific traits were averaged out. So in the future, I would like to do more than only linear models, but use mixed models, for example, and put species in as a random factor to take this into account. And also include non-climate stressors if that data would be available. As example, fishing pressure, habitat degradation, invasive species, because besides from climate metrics, these also really would have an effect on the redistribution of species. So the 5 major conclusions are that redistribution is a basin-wide phenomenon, because in all 30 of the subregions and in all 3 of the spatial dimensions, there were significant shifts detected. The meridionalization theory is only partially supported, and then the Mediterranean demersal redistribution is a multi-scale climate response, because we saw that both the seasonal acute and chronic processes were taking place at the same time. The seasonal metrics were the most pervasive drivers because they were retained in all 3 of the models. And then for the management applications, I would suggest more regional conservation framework instead of the basin-wide generalization, because as we see, each of the subregions have different movements of the species, so it would not make sense to make it more general across the whole basin. Thank you all for listening, and a special thank you for my 2 main supervisors in the Centro Oceanográfico de Baleares, one of them might be looking funny right now because he was not originally included in the picture, so I had to Photoshop him in. But yeah, big thanks to Manolo and Marina and everyone else that helped me in this thesis presentation. Because I think it was all quite clear, there are no further questions. Yeah.
Thanks very much, Louise. Well done. Very nice presentation. Interesting topic, a bit to get my head around because it's a different topic to what I do.
No worries.
But let's go for it. So you mentioned at the very beginning these 30 ecoregions.
Yeah.
Who came up with those, or was that in the published literature, or?
Yeah. So a part of those, like previous studies have already been looking at the Western Mediterranean. So 15 of those subregions were already made, but the Eastern Mediterranean was still quite new to discover because it hasn't been done on this scale, and there's also more information available of the Western Mediterranean because of funding reasons and stuff like that. So it was quite interesting to also have a look at those regions and see if they have similar results as well. I had a look at previous studies and applied the same polygons to include in R.
And then I was wondering, in terms of you then looked at different shelf versus slope habitat as well.
Yeah.
So I was wondering if there was overlap. I know it wasn't mentioned in the collinearity or anything like that, but I was wondering if there was overlap between the habitat and the ecoregions, because you might expect the species to respond to that, or did you look for that or test it?
That would be really interesting to look into the future as well.
Okay.
I didn't check whether or not the slope or shelf habitats had like a major effect on the redistribution patterns that we saw. But obviously it would be something that would have an effect on.
I think it's interesting because you said the ecoregions were decided by the biodiversity kind of in that region, right?
Exactly.
And that's probably affected
A major effect on it as well. For sure. The topography of the seabeds would be something interesting to include as well in the model.
And then another one was the 40-degree north line. Was this what was the relevance of that, or why do you think that's come out?
This was one of the major findings I think I did in this study, and I mean it's really difficult to find patterns in these graphs, but this was really one that stood out for me, and to compare it to like the expectations of the more poleward redistributions, it's something, and I don't have an explanation why it exactly is.
Like what would be kind of the middle of the Mediterranean?
I mean, not the Mediterranean, because also like the southern
that's what I mean.
regions haven't been taken into account because this is more European study, so I cannot claim it to be a Mediterranean study entirely. So I decided to put this one as a division line, to better explain the redistributions that we saw.
And then if there's time, like I still have one or two more, but I'm happy to be cut off anytime. I was wondering, so you mentioned how the east-west is kind of something new to be looking at, and I think it's interesting, but I don't know why I think it's interesting. So I'm wondering if you can explain a little bit what you came across in trying to figure out why you look at longitude and what might be driving these east-west. Like what will it differ in different environments? You know, you talked about how the Western and the Eastern Mediterranean are quite maybe different, or one is more exposed to climate.
Exactly. We
extremes.
can have a look maybe at the maps I created, because you can clearly see that the Eastern Mediterranean, for example, for the long-lasting warming trends, is more exposed towards warmer sea surface temperature. So here you can see a clear gradient that we also saw in the partial residual plots. So species are really responding also towards more intense climate change effects. And I mean, this is quite homogeneous, so the previous studies don't really show like a difference in this. But longitude studies, I've had a look at a few papers and I didn't come across many. Only latitude is one of the major factors that they're studying nowadays. So
I found it interesting that you picked up on this, you know, within the Mediterranean, because I think it
Yeah.
might be quite environment specific, and you know, if you start to compare across
Yeah, for
the
sure.
North
Exactly.
Atlantic or something, it might be too big, but where you know there's extremes within the environment already, that was kind of what I was wondering. Yeah. Okay. Thank you very much.
Yeah,
I'll stop there.
Louise, thanks very much for your presentation. Very nice. Well done. So, would you be able to elaborate a little bit on the depth, because you found kind of opposing results.
Yeah.
Would you be able to go a little bit further on maybe perhaps a tentative?
Time to explain.
Yeah,
it.
yeah.
Yeah, that would be something I would love to know as well. But yeah, it's if you have a look at this map, it's really difficult to try to make up like a reason why in the Western Mediterranean there are more shallowing trends taking place than in the Eastern and the Adriatic Sea. I think I have to read more into literature and to see if other studies have also seen like similar results. For example, the one of Sans Martin also came across this result, which is opposed to the meridionalization hypothesis, but to really come up with a reasoning why this is, I'm also lost for words for now.
Fair enough. Do you think, like in terms of the way that you analyze the data, if you had to analyze that again, like would you do a little bit of more streamlining in terms of like species and that kind of stuff?
Yeah, for sure. That's something I'm really looking into the future, because for now it was just simple linear regressions, and we didn't take really like species into account. And as you may know, like species, all of them have different thermal tolerances. They have different dispersal capacities and body weight, body size. Like, I think the first step is to deviate the bigger taxonomic groups and to have a look into which each and every one of them are moving to. And then if I have more time and of course more resources, it would be nice to see species-specific differences as well. And for example, have a look at what, like one of the most commercial fishes that were found in this study was the European hake, Melucius melucius. It would be nice to see if like the redistribution of the species in the Gulf of Lion is the same as in the North Adriatic Sea, like, and compare and find reasonings, why that might be. So yeah, in the future for sure, I would like to make the mixed model and then put that as a random factor to better or more specifically look into species as well.
Okay, perfect. Just one more. In your conclusions, you mentioned that one of your conclusions is that you can have like management actions like generalized for all of the different, regions. So what kind of management actions are we talking about here?
We could have a look at if the species are also decreasing or like increasing in a certain area, because the MEDIS data set has been specifically made for commercial species as well. Like, this is the primary reasoning of creating the surveys, but yeah, I'm grateful that I can have a look at all the species that they picked up on. But yeah, for example, for a commercial species, if they decrease, we can make management actions to make sure that the population is increasing again in the future. But also more in general, like this study really shows that climate change effects show that not only in the future but already nowadays species are reacting to it. So I think it's just maybe more a general study to wake up policymakers and to take actions into avoiding even more worse conclusions, I guess. I don't know.
Okay,
Sorry.
thank you. Okay, thank you.
Congratulations, Louise.
Thank you.
Really impressive work, complex, and you did excellent to show it and explain. Extremely relevant. There is more. Yours, as we are, you will address, you have been addressed by my colleagues in the discussion. You did already a lot of arrives and managed to show very important results in terms of extremely relevant to test this kind of hypothesis that already. I know that in some levels they are being taken for granted as in the Mediterraneanization, as a for sure. What I find very interesting of your conclusions, the need to go on a smaller scale to investigate things and to refine it. But did you have the chance to reflect, and if so, what would be the first steps to try convincing, especially those that are funding projects, so not only because I think it's easier to convince the scientists, but when people, not only the policymakers, but the funding agencies, have so much a word and a need, that's what I would guess, but feel free to disagree, but that's one of the challenges to say, no, we don't want to go in that scale, you need to create opportunities to fund smaller scales projects to understand what is happening.
Exactly. I don't think this is the only study that proves that ecosystem-based approaches are more important than looking at, for example, the Mediterranean as a global unit. So in the future, it's already like this study shows that like combining all of the European countries really has like an effect on, like showing what is going on in the Mediterranean. But to have the specific question of you is to
how, if you have the chance to think, how would you start convincing what is needed to convince? Okay, this is the result, but what would you have the chance to
keep on going like this project, or to have like
now let's say I'm a funding agency, but I have the mindset that okay, I have money for the Mediterranean, you want to focus on one specific region. Well, did you have the chance? How would you convince me further? If I'm not a scientist, so it would be difficult to understand the details of it.
I'm not really sure. That it's policy making.
But would the local biodiversity be a way to help to reflect on locally and with economical activities, the importance of it?
Yeah, for sure. I think those are already good suggestions.
Don't worry. I know that that is not the core. This maybe on the shift on the variables or the model. Maybe I miss it, but were the currents considered, or how much they would consider, could they be considered in addition to explain the trends for temperature, for example, but also for nutrients, that further adding for us to understand the shifts on the species.
Yeah, exactly. Yeah, it would be super interesting. I did read a lot of the currents being added into a model depending on demersal species, but I can give it a try in the future. But I know working with currents is quite difficult also to do. But it might be a possibility for sure. Thank you.
Okay, thank you, Louise. I really enjoyed your talk as well. Very clear and everything. But there's one thing where, so one of the big takeaway you give from your talk is that this meridionalization hypothesis might be only partially true. It's like not perfect. And there is one thing on which I would be perhaps a little more prudent, but I don't know if you thought about it. Have you included in your thinking on, I don't think in your analysis, but in your thinking, the issues you can have with whether a species is as its leading edge or trailing edge in its distribution? I don't know if you're familiar with that.
Yeah. I've also looked into that. But that would also be like one of the future perspectives, I guess. Given of course the restricted timing we have here. But it would be nice to for the future.
Do you see how that could possibly affect your conclusion if you include that? Do you have an idea?
for now, I don't really know because I haven't really read into that kind of literature before.
And to be fair, that might explain some of the pattern. I don't know because I haven't looked at the data, but that might explain some of the pattern you have.
Okay.
Sometimes the issue you have, especially if you are at the leading edge of the species, that you're only gonna find a few individuals, and when they start to move north, then in the south of your region, you start to have a lot of them, and so the symptom moves southward, but the species is moving northward. So that's a potential confounding factor, I guess. That would be great to have an idea of which species it's where in its general distribution, so not only in the different areas. So I guess that was a main question. I don't know if I have time still. No. Oh, I can go. I'm sorry, she allowed me. I had another question about the way space was included in the models. So, so for instance, you ended up removing longitude because it was correlated with other stuff. Do you know with what it was correlated?
Yeah, the model doesn't really specify that, but I assume that it's going to be with, like the other longitudes.
Yeah.
You really see a clear pattern also with the chronicle one. So I'm assuming it's going to be that one. Yeah, it's a pity I couldn't include that in, but I mean, otherwise the results wouldn't have been valid. So yeah, that's the reason why I had to take it out as well.
So it's only on the basis of that collinearity that you remove them. Okay. Yeah, that's going to be a technical thing. Okay. On this specifically on space, do you see an issue with possibly only including latitude and longitude like that, specifically for space in the linear model?
I'm not sure what alternative approaches would be.
A lot of them.
So yeah, no, for now I don't think I see the problem with that.
Okay. I guess it's not necessarily a big problem with the things that you only model a trend.
Yeah.
And if you, for instance, it looks like it a little bit in your depth deepening or shadowing movement. It looks like there is a bit of a hump, like in the middle of the area. There is like bigger effect than on the side.
Yeah.
And you cannot capture that with only a trend. And with space, you can really have those multi-scale effects that are not represented by your only student.
Okay. That's interesting. I want to have a talk with you.
Yeah, I mean, you can. I think I'm critiquing now, but I think there's a lot of really nice work that you did, so it's not.
Okay.
I still just 1 minute.
Stepping in.
You realize until 2021 because there are no data more on this, or just
there is but we decided to only work with this data because afterwards.
Okay. So they are still continuing the.
Yeah.
Do you think that is a sustainable way to collect the data since it's rolling?
Yeah, I wish there were, but as we all know about controlling surveys are one of the worst of the worst. But it's for scientific research, so it's okay.
So now we are looking forward to new technologies, so maybe a shift in that.
But yeah, for now they're still continuing, I'm afraid. Sorry to disappoint you.
No. It's only for thanks a lot. Okay. We finished? Okay, thanks.